Anshu, are you able to hear me?
Yes, I can hear you.
Okay, fine. Yeah, just wanted to confirm that. Yeah. Yes, we will wait for another 3 minutes. Um,
Yes, Kiran, tell me.
yeah, we will wait for another 3 minutes. Looks like people are still having lunch, so I will wait for another couple of minutes.
Sure, let me know when we
Yeah,
can start.
sure. Santoshi, have you sent to everybody all the list of participants?
Yes, sir. Yes, sir.
Okay, thanks.
Thanks.
Thank you all. So after the lunch, we have another workshop session, and this session is headed by chaired by Anshu Bhartwaj, and I'll give it over the session to Anshu. Anshu, please take over. Thank you.
So, thanks a lot, Kiran. And I think we do see a few people online, so let's get started. So, a very warm welcome to WS4 of the INBIX 2021 Pre-Conference Galaxy Workshop. And in this session today, it's my pleasure to welcome Sona Charles. She is a scientist in agricultural bioinformatics with the Indian Institute of Spices Research. Her areas of interest are next generation sequencing, genomics, gene networks, and systems biology. She also has interest in applications of machine learning to molecular biology. In today's workshop, titled Proteogenomic Workflows Using Galaxy, she'll be demonstrating workflows in Galaxy platform for proteogenomics analysis. You all know that proteogenomics is the boundary between proteomics and genomics and involves the generation of customized protein sequence databases that facilitate the identification of novel peptides obtained from mass spec experiments. Now, this approach has led to better annotation of protein coding genes, and it can also help us identify and characterize novel peptides and provide insights into their biological functions. So, without further delay, I again, on the behalf of the INBIX organizing committee, invite Sona Charles for her presentation.
Thank you, ma'am. Thank you for the wonderful introduction. And first of all, I would like to thank the committee for selecting me to give a presentation in this platform because INBIX is one of the greatest bioinformatics conferences in India, and I'm very happy to be a part of this. So, as ma'am has told, today I'll be talking about the proteogenomic workflows using Galaxy. So, before going directly into the workshop, I will just give a brief outline of what proteogenomics is and how it can be done and what are the data files that you will need for running the workflows. So, I'll just give a brief presentation and then we'll go on to the hands on session. I'll share my slides.
Yes, you can go on now.
So, I hope the slides are visible now.
Yes,
Um,
yes, Sona.
Okay.
but Sona, they are not in the presentation mode.
Is it not? Actually, it is in what I'll just do it once again. Is it okay now?
No, you have two monitors.
No, I have one. I'll just stop and share once again.
So, Sona, do one thing.
Yes.
Make it into a presentation first, in a presentation mode, and then shift windows. Hopefully, it will work that way.
Okay, fine. Now is it fine?
Perfect.
Okay, fine. So today I'll be giving a brief introduction on proteogenomics. So we all know from the title itself, we all know that proteogenomics is the interface or it is the combination or the integration of proteomics with genomics. So it all begins from the basic central dogma of molecular biology, where we know that we all study it in lower classes itself. So now we are applying all those techniques, and the central dogma states that the DNA is converted to RNA by the process called transcription, and RNA is converted to protein by translation. So everything is actually running on this basic central dogma that is actually the entire process of life itself. And now we are trying to find out new methods to find out what exactly is happening at the DNA level, the RNA level, and at the protein levels. So proteogenomics allows the correlation of the microRNAs, not the microRNAs, the mRNAs and the proteins across different samples to evaluate the mutations, then the post-translational modifications, then the signaling pathways, how they are affected in response to a condition, and the correlation between different regulatory effects of the RNAs and the protein expression levels. So basically, the data that we obtain from proteomics is by the mass spectrometry or the MSMS. So what exactly happens here is we have proteins in the body or in any other cell or in the condition, whether it is in plants or in the human body or in the microbial system. So all the proteins are first digested using a set of enzymes like trypsin, chymotrypsin, etc., and the proteins are fragmented to peptides. So the peptides are identified. They are identified using the liquid chromatography, and then they are ionized and they're given charges. And these charge ions are then isolated and they are fragmented, and finally they are run through a mass spectra analyzer where it is converted into peptide fragments, and it is arranged, or it is made in the form of an array of molecules with increasing charge and mass ratio. And it is ultimately converted into a form which is called the spectrum using a detector. So the detector is coupled to a computer and we can read the information through the computer screen. So it is basically like from a complex mixture, we are separating and detecting and identifying and studying the features of each of the components of the proteins. So it's a very unique technology, and we can expect that a lot more things can be done using the proteomics data. Now, once we get the raw MSMS spectrum, we feed it into the computer and we search it against the protein sequence or the DNA sequence database. So as we know that the database sizes are getting very bigger, so the search is also very comprehensive, and we have a certain theoretical sequence, the theoretical spectra that can be matched to the spectra that is obtained from the MSMS. And if it matches, we can say that it is similar to that one in the reference database, and if it is not similar, we can say that it is a novel peptide. So it is not always the right way to look at the ones which are already available. So we have to look a little bit further beyond to know the proteome. That is, we have to see what is there in the mass spectrum that has not been detected yet. So in such an approach, we sometimes will be able to identify novel proteoforms of the proteins. So when the novel proteoforms is combined with that of the genomic and the transcriptomic data, finally we will be able to find out what is missing in the there that corresponds to different conditions or different treatments. So this is exactly the mechanism in which multi-omics technologies work. So here in proteogenomics, we'll be combining the DNA level information, the RNA level information, and the protein level information, and finally identifying novel peptides that may be contributing to a particular condition. So the concept which I have explained is very simple when we are seeing it, but still, if you want to read more about it, I would suggest you to read this paper. This is Proteogenomics Concepts, Applications, and Computational Strategies. It has appeared in the Nature Methods in 2014. It's an excellent paper that describes about what proteogenomics does and what all information can be obtained from that. So I suggest you students to read the paper, and now we will just move on quickly to the steps so that we can directly go to the workflows. So basically, the steps in this proteogenomic methods are, the first step is like we will get the raw RNA-seq data. So that comes usually in the form of FASTQ files. So the FASTQ files are processed and a
Excuse me, Sona.
protein. Yes, sir.
Uh, can you please click on that hide button so maybe the complete slide will be visible?
Okay.
Great, thanks.
Okay. Now, the RNA-seq data is processed, and it finally will get a protein sequence database out of it. And then by using the raw MSMS data, we'll be searching the database. So we'll be combining both of the data from the RNA-seq as well as the MSMS, and then we will be searching against the database, and the proteins or peptide are identified. So once we have identified the proteins or peptides of our interest, or if we have identified a novel one, it is very important that we should visualize the results. So another option is therefore visualization called MVP viewer, where we can visualize all the results that we have obtained from the pipelines. Now, so this is one point where we have to go into a little bit deeper. So our entire proteogenomics workflow is consists of three main workflows. So the first one includes the all the information regarding creation of a database. So the most ideal way is to go through the workflow in that order, and then you will understand which all files we'll be using for the next upcoming steps. and in what order we'll be using that. So in the workflow one, what Galaxy will be doing, the workflow will be doing is, they'll be taking in the genome, the reference genome, then the RNA-seq FASTQ files along with the GTF feature files. So the reference genome with the reference genome, the RNA-seq FASTQ files has to be aligned, and this is aligned by using the HiSat tool. Now, for assembling the transcripts, we need the aligned reads from the HiSat, as well as we need the GTF files. So we use string type to do this assembling, and we need to see which all transcripts are there and which is not present in the genome, or which is present in the genome and is not present in uh, the FASTQ files. So we have to run a GFF compare tool to compare the annotations, the features, and then we have to translate them, the unique ones, into the corresponding transcripts. Now, there is another option called the freebase method here. So freebase is a Bayesian algorithm based a tool that helps to identify variants, or the SNPs, or the single amino acid variants, or any indels, or insertions or deletions. So we'll be running freebase also. And finally, the result is then given as an input to the custom ProDB for genome level mapping. So at the end of this workflow, we'll be getting the map files and the sequence FASTA files. So this entire workflow will create a database for searching, and we'll be using the results from this workflow to go into the next workflow. So that the second workflow basically deals with creating a list of peptides for BLAST-P analysis. So RNA-seq database is combined with the MGF files, that is the proteomic data files. So initially in the first workflow, you can see that we have used only the FASTQ files. Now in the second workflow, we'll be using the proteomic files also, and then search GUI will be performed, and then it is given to a tool called the peptide shaker, and finally the MC to SQLite 5, and we will get the peptides for the candidate peptides that are suitable for a BLAST-P analysis. Now, the third workflow takes in the input of the BLAST-P, the peptides from the BLAST-P, and it does a NCBA BLAST-P on the peptides, and then we have a PSM matrix, and in the PSM information, it contains the novel peptides. So the novel peptides are then taken, and it is mapped against the genomic coordinates, and by using tools called the pep pointer, annotation is made, and finally we'll get a summary information of the novel peptides. So this is the overall outline of all the workflows that we'll be using today. So I think we will directly move on to the Galaxy tool to do it. So I think everyone will be doing it with me, right, Kiran sir? Shall we just move on to the hands-on?
Yeah, sure, Sona. Go ahead, please.
Okay. So here, I would like to show you, before going into the hands-on session, I would like to show you a few things. For this, we have a detailed tutorial that is created by the Galaxy training team itself. So it has 3 tutorials. So I shall share the links of these tutorials to you. And here you can see that is we have 3 separate tutorials for each of the 3 sections that I have described. And if it is convenient for you to directly copy and paste the codes from here, paste the lines from here, then that is fine. But I think it will not be completed before the prescribed time. So we will do one thing. We will just import the already created history that is there in Galaxy, and I will be explaining each and every tool, each and every step in the analysis, what each step returns, how the file differs and all. Then we will move on to how to do it according to the tutorial. So first, I hope everyone is signed in to Galaxy, and I will be telling you from scratch on how to start. So you have a shared data menu here. So you click on shared data and you click on the histories. You can search for proteogenomics. So here we have so many shared histories, but right now I want the one that is created by Galaxy P itself. That is Proteogenomics 3 Novel Peptide Analysis. You can click on that. And you can import the workflow. So it's taking a little bit of time. Just wait. Okay, so this is the history that has been created by the Galaxy P team, and you can directly import this history into your account. Just click on import history. You can just rename it Proteogenomics 3 novel, okay, you can just give a new name, INBIX Proteogenomics. You can import this work in this history. So if it is imported, it will be shown as a new history in your account. Okay. So here I have the history imported. So from here, I'll be explaining what each and every tool does and what each and every file stands for. So I'll go a little bit slow from here so that if at all possible, I can give you some time for the hands-on session also before the end of the workshop. So here, if everyone has imported the histories, you can find here that the first file is the FASTQ file. You can just click on it, explore the data. So basically this FASTQ file which we have imported is the information regarding uh, the B cells under different conditions, the different developmental stages of the B cells. So the information is given here in the tutorial. So if you want, you can just read more about it here in the original reference paper. So I have imported that FASTQ file. So another, I hope you're aware of other ways of importing. Like here, if you are going to follow the tutorial, you can see that they have provided the links for all these files. The FASTQ file, then the features file or the GTF file, and the FASTA file. So if you want to copy it directly to your Galaxy account, you can just click on copy. Then you can go to Galaxy and you can just click on upload data and paste or fetch the data, and you can paste it right here. So since we are already dealing with one history, I'm not going to show you how to do it, but this is how you have to follow if the history has not been imported. So next we have the GTF file. So if you click on the eye icon, you can just view what is there in the GTF file. So you can find out all the information here because it doesn't make much sense since it is just clumped up. We only want the relevant information, so we can just try to find if we'll get some novel peptides after running this workflow. Now next we have the FASTA files of the protein, FASTA file of the proteins, and after that we will be moving on to another tool, that is the HiSat tool. So if you have worked with transcriptomics, you most probably might have used the HiSat tool. So HiSat is used to align the files to a reference genome. So here, if you click on here you have a read on button. So if you click on this button, you will be able to see the parameters which we have given as input for running this tool. So here, uh, We have given a built-in genome, that is musculus 10, for this analysis. And since we have only one FASTQ file, it is a single end file. So, and we have given the name of the FASTQ file here, and strand information is unstranded. So now after this, you can just click on execute. So if you want to run this workflow along with me, you can just follow the same, you can use the files from the history and you can just click on execute. If you want to rerun that again, it will create another file on top of what it is already there in the history. So if you want, you can try it out. It will take a little bit of time to run, so you can just wait until it is run and see the results. So once you do HiSat, you will get an alignment file. So basically, when we are using Galaxy or any other tool, the idea is to format the files in such a way that it is compatible with the tools that are supposed to be used downstream of the analysis. So that is exactly what we are also doing here in Galaxy. So the only thing is that you should be able to know what tools are there in Galaxy. Like if you see on this tool in the Spain tools pane, you can see that so many tools are there. But even if you ask me also, I am not aware of what all tools are there in different uh, on different kinds of tools. So there are so many tools which you can use and you can just try it out. Even if you get an error, it is okay. You can always try it out from the tools pane. So then only you will understand what are the different tools that are there and what exactly it does. So this kind of logic is required to even create a workflow using Galaxy by your own. So that is very essential. You should be familiar with all the tools. Now, the next step is exactly what I was saying. So next step is I have a GTF file by the name edited musculus. So we will just see what has been done to that file. Like we have only 3 files in the that has been imported. Now whatever files that are being created is created from the 3 imported files only. So we'll see what happened here. So here the file to process is the musculus GTF file, the one that has been already uploaded here. And so some replacements have been done. So what exactly are we doing this step for? So here I'll just show you this file once again. So this first column, you can see one, and if you go below, you can see the chromosome numbers. They are just given as numbers. So but this is this kind of GTF file is given by the ensemble. So right now, for our analysis, we do not need the ensemble format. What we need is the UCSC format. So we need to convert the ensemble formatted file to that of the UCSC formatted one. So we will see what we can do. So here, for getting this edited musculus, what we have done is, here you can see in the first column, one pattern has to be identified and then it has to be replaced with another term. So this is nothing but the regular expression pattern. So if you must be familiar with the regex and regular expressions. So if you are not familiar with how to use it in Galaxy, you can just go down in the tool itself. They will give you the exact meaning of what the pattern, what the pattern is, and the meaning of the pattern. And you can also see the regular expression syntaxes, and you can identify what it is. So right now, what our regular expression does is, we have 3 regexes. So what it does is, it finds in the column 1, it finds a pattern with the numbers. So instead of the numbers, it will be giving the chromosome prefix, CHR prefix. So that is the format of UCSC files. So likewise, the X and Y chromosomes has to be replaced, and similarly the mitochondrial ones also has to be replaced. So if you click on execute here, it will again run and it will give the result. So I'll just view the result. See, earlier the file had only one, now it has a prefix of CHR. So this is exactly the way we want it in the UCSC file format. Now, next again, we are going to play with the data. Actually, that's what I would like to say, because we would like to format the data in one way or another, and ultimately we want to make it in the way we want to for giving as that it as input to the next tools. So here we have a tool called the FASTA to Tabular. So if I click on run, so here what they are doing is they are taking this FASTA file, and it is dividing, it is separating the FASTA file into different columns. So we will see what it does. We have now two columns. This is the first column consisting of the name and protein name, the accession number, and the details regarding that. And in the second column, we have the protein sequence. So this is what FASTA to tabular tool does. So it splits the FASTA into a tabular file, and the second column consists of the protein sequences. Now, earlier in the workflow, you must have noted the tool Freebase. So I'll show you what Freebase does. So the input file for Freebase is the aligned, the HiSat to aligned files, the BAM files, and with the inbuilt reference genome MM10, and it identifies the variants when the aligned reads are compared against the reference genome. So if you want to know more about it, you have the option to read about it here. So Freebase is a Bayesian genetic variant detector. It's used to find small polymorphisms, then SNPs, indels, then multinucleotide polymorphisms, and insertions, substitutions. All these things you can detect using Freebase. So it actually gives a VCF file. So this is the content of the Freebase result. See here, you can see the chromosome number, the position, then what has been changed, the quality, filter, all these details are here. Now, in the next step, they have used string tie. So what is string tie? String tie is used to assemble the transcripts. So here, if you see the job information, you can see that it has taken in the BAM files of HiSat2, and the data set that has to be guided for the that has to guide for the assembly is musculus edited one according to the UCSC format. Then you can just click on execute to run this file. So what we get here is the assembled reads. So they have mentioned whether it is a transcript or an exon, and the location, then all the other information regarding the transcripts, then the FPKM values and the TPM values. So many information are here. So that is what we get when you are running the string tie tool. Now, next step is again, we are going to run filtering. So here, earlier we had done one FASTA to tabular tool. So on that result, we are again running a filter tabular to filter out certain fields. So what are the different fields that we have to filter out? So we have a regular expression pattern. Based on that, we are filtering. So we will see the output of that. So here, based on the regex pattern, it has returned us with the following result. So these are just the accession numbers of the proteins of the reference. So earlier we had the accession number along with so many other details, and then came the sequence. Now we just have the accession numbers. Now I will introduce you to a new tool, that is the custom pro DB. Now next step, we'll be using the custom ProDB. So here, as the tool mentions, custom ProDB generates protein FASTA from exosome or transcriptome data. So that is exactly what we want. We want to get the protein FASTA sequences from the transcriptome, the FASTA files which we have earlier imported. So how to do that? So here also the genome annotation file is taken from the inbuilt file, and we have the BAM file, and we need the VCF file that has already been generated using Freebase. So once you run custom ProDB, you will get a few files. So I think you will get about 8 or 9 files regarding this custom ProDB results. So if you want, you can just click on execute and run the step. Now here you can see I sat on data 1 align reads, that is rpkm.fasta. So it's a FASTA file. So all these are FASTA files. Then SNV, single nucleotide variations, then indels, then variant annotation and R data. Then we have a mapping SQLite file, then also an annotation SQLite file, then the assembled transcripts. So we have this much. Now so if next step is we have to run a tool called the GFF compare. So GFF compare, as I've already told in the previous PowerPoint, it is used to compare how our novel, or not the novel, how our assemblies differed from that of the original genome assembly. So for that, so we are giving the string tie on data 4 and 5 and 4, the assembled transcripts. So that is our set of assembled transcripts, and we have to compare that with that of the reference annotation. So here we are giving the reference annotation file, and you can just click on execute. So here GFF compare will be giving two sets of results, that is 16 and this one 18 with two files inside. So we'll go on to the next step. Oops, actually this I have already explained. So this one also I have explained. So GFF compare will be giving so many results. So here, until here, we have GFF compare results. So it gives so many results like the annotated transcripts, then the tracking files, then the loci, then the transcript accuracy. So many information are there. So we'll be using some of these files later in the workflow. So you have a SQLite to tabular file here. So let's see how we got it. So SQLite to tabular file, it is mainly for querying a file. Like we have a SQLite database file that is already here. We have obtained this result earlier, and we are giving an SQL query. So as per the query, we have to select certain columns, and we have to order it by certain in a given order. So this is done in this step, and it's a tabular file. I don't think I can view it. Yes, I can view it. So this SQLite to tabular will give results in the form in which we have asked it to give according to the SQL query. So this is the resulting file. Now we have we have so many FASTA files that have been generated from custom ProDB. So in this step, we'll be merging all those FASTA files. So you can just give a rerun here. So what are the files that are we going to merge? So first thing is we are going to merge the align reads rpkm FASTA, and then we are going to take in the align reads SNV FASTA, and again we are going to take in the align reads indel.faster. Okay. So this is done using the tool FASTA merge files and filter using unique sequences. So this is for in this workflow, this tool helps to combine all the FASTA files together. So we'll just see the results. So it is a combined file. I don't think you'll be finding any difference here, but if you check on the total number of sequences, you will be finding that it includes all the sequences from the previous list, previously mentioned files. Now, again, we are going to query the files. We will see how it is done. SQLite to tabular on data 15. So here we have the HiSat2 variant annotation SQLite file. This has been previously generated. Here, variant annotation file. So here, what do we want to query? So we want to select all these items from variant annotation file. So if you click on execute, it will be running in the background. We will see what it gives. So now this is the file containing information in that order. Now again, we are going to do some more manipulations here. Pasta to tabular. We will see what it does. So here also we are taking in the rpkm.fasta and then you are converting it. You have to divide. It's mentioned that the columns to divide the title string is 1. So ideally it will give a result. I'll show you here. So this is the kind of result that will be obtained after this tool runs. We will get the sequence and the information regarding the sequence in two separate columns. So only once we reach the end of the workflow, we will be able to understand why we are creating, why we are again doing FASTA to tabular. So it is basically to bring the data to a format that is most compatible with our final tools. So that is why we are running all the tools. Now, next one is we have to convert the GFF compare annotated GTF to a bed format. So we know that the bed file format consists of information regarding the chromosome start and the ending regions. So we will see what it does. So here we do not want all the transcripts. So we are actually looking for novel transcripts in this workflow. So we do not want all the transcripts. So now we are going to find out ways to filter them. So the first, here are some parameters which we can use to give a cutoff, like we want the potentially novel isoforms or the fragments with at least one splice junction is shared with the reference transcript, then the single exon transfrag overlapping a reference exon, then a transfrag falling entirely within the reference intron, then possible polymerase run-on fragment, then unknown intergenic transcript. So if we give these cutoff values, all the transcripts will not be included. So we will be getting a summary of the transcripts. We will see what it has written. So here we have only four transcripts. So now column regex find and replace. So again we are trying to find and replace. I will show you what the tool does. So here we are selecting the cells from the SQLite to tabular data on data 14. So I'll show you that. 26. So this is the file which you are going to use here, and again we are using some regular expression to filter it. So here you can see we are trying to remove the ensemble and replace it with something else. So we will see how the file will look like. See here, so the information has been modified in a different way according to the input. Now again, we have to do a FASTA to tabular on the merged custom DB files. So remember that the custom DB files have been merged into one single file called the custom ProDB FASTA. So all the different FASTA files from the RPKM and the variant information has been now combined into the single file. Now we are also running another FASTA to tabular on this, so as to separate it into the right way in which we want. So we will see the result. So it will be like this. It will be separated into two columns. In the first column you'll get the name, and the second column you'll get the sequence. So again we are doing the same thing, but this time we are doing it on the SQLite file. Again we are doing some regex here, then click on execute. So this will be returning the file in this format. Now again we are doing it on the GFF compare. So finally the parameters that have been used here are the same as before, like we are giving some regexes and we are filtering the tabular to FASTA on data 10, this file 29. So this is the file which we have chosen for this, and you can click on execute. So after the step we will be getting all the ensemble IDs. So now I think you have understood like why we are doing each step. Now we want to make the data in this format. That is why we have run the previous steps, so that we can filter the contents of the file at each step and finally make it in this format. Now we have translate CDNA minus CDS on data 26 and convert GFF compare annotated GTF to bed on data 1. So you can just click on that. You can just click on run. So here we are just translate the bed transcripts into the 3 different reading frames or the CDS. So for this we are using the GFF compared data, and the reference is MM10. So not much parameters have been modified from the default, and you can just click on execute. So both these are the results of the same tool. So here you can see that we have the sequences and the names and the chromosome related information. So before moving on to the next step, I would like to tell you one thing. So here you are mentioning the FASTA ID or the FASTA source as generic. So whatever we suspect it to be a novel one, we'll be giving the term generic so that it can identify. Like we are giving this input into another tool called the peptide shaker, so it will identify the term generic. So that is why on the result file here the term generic is coming. So it consists of information about the string type ID, then what kind of the exon or intron or what kind of transcript it is, and we have given the option J, so it stands for that it has got a meaning, and here you have exons and here you have introns. So all these information will be coming here. Now again we are running column regex, find and replace. So this time what are we going to replace? So we are just going to split it into different columns based on different regexes. So here we are replacing it with generic, and we will see what this file returns. So here you can see the beginning of the file. This the file itself begins with generic. It has got two columns, one with the sequence and other one with the information. Now we will move on to the next step. We are going to do a query tabular here. Got this. Again, we are using a query tabular using SQLite. So on what data are we using? We are using the column regex which I have already created here, that is 33. So that is done on the variant annotation file. So already we have created a regex on that, and again we are going to query it. So here we are giving some options for filtering. So what are the columns that are needed is mentioned here. Then so accordingly it will modify. We don't have any query yet, so accordingly in that order it will be modified. So it is a SQLite file. Now in the next step we will be getting the reference protein accessions. So how are we getting these accession numbers? So we will be concatenating the data sets. So earlier itself we have filtered and we have got two files that has got the accession numbers or the uniprot IDs. So one is the filter tabular on data 6 and the other one is, I think I missed it. Just rerun it again. Here filter tabular on data 25. So we have to concatenate these two data sets, and finally we will get a list of reference protein accessions. So we now have the whole list of reference protein accessions. So these are all parts of steps in making a database. Now we have a tool called the bet to protein map, which is the translated file. Yeah, it uses the translate cDNA minus that file. So it is used to map the genomic location of the proteins, which will be further utilized for our MVP visualization. So you can click on execute. If you view this file, here you can see the string type IDs with the information regarding the chromosome number, the start and stop region, then the strandedness, then whether there are any coding sequences, and if it is there, at what region it will start and at what region it will end. So all these information will be coming here. Now next we will run the tabular to FASTA on the variant annotation file. So again we have to modify our file. So the column that is selected is one, that consists of information regarding the protein, and we have the sequence in column number 2. Now we are converting the FASTA files that have been converted to the tabular back to the FASTA format in the right way. So we will see the result of after running this. See, this is the FASTA file. So it is just like we are converting a FASTA file into a tabular file, or you can imagine this in such a way that you are copying your file from a notepad to an Excel sheet, doing all the manipulations in the Excel sheet, like you are pasting it into different tabs and you are editing it, you are adding new values, and again you are turning it back into your FASTA file. So that is exactly what we are doing in Galaxy in this workflow. Finally, we need a database. Now we have the GFF compared and the bread and the bed file and the protein file. So we need to concatenate the entire data sets. So here you have to just keep an eye on what file you are going to use for each step, because there itself sometimes you can have some confusion and it will not give the result. So here we are using the column regex on data 22, and then bed to protein map on data 31, and we are concatenating. So here you can see the results. So this is the final result of the file. So now so earlier we have uploaded one trimmed reference 500 uniprot reference file. So we are going to merge the FASTA files and filter the unique sequences. So while merging, we need this file, the trimmed uniprot reference file, then the tabular to FASTA on data 33, then you need the translated file also. You have to merge it. So the parsing regex and all is already default, so you don't have to give it there. So you just have to mention the file names and we will see the result. So it consists of so many sequence information. So this consists of the single amino acid variation, then the indels and the translated bed sequences. So we have the proteins. Now, the next step, again, we are going to use the query tabular using the GFF compare and the custom ProDB. You will see how it is done.
Sona,
Yes, ma'am.
maybe we can actually stop for 1 minute and see if anybody has a question or if they have any feedback. And maybe you can also, just show the original workflow one more time
Okay.
to put in context of how far you have reached, so that there is some alignment with the process and the steps, so it will be easier to follow that way.
Okay.
So let's see if anybody has any questions so far, and then we can move forward.
Yes, ma'am.
So if anybody has a query, please feel free to let Sona know about it. And if everything is going fine, we can wait for a minute. If everything is all right, then we can proceed. So we'll just wait for a minute for you to ask any questions. Since it's a workshop, it's important that you are following what is being told. Okay?
Yes, yes. Any questions? Or, ma'am, shall I just complete the first workflow and then give a few seconds to just go back and see what has happened? Like you have only 2 steps more to complete the first workflow.
Yes. So you can finish this workflow and then go back to the slides, and we can come back here.
Yes, yes. So, okay. Now we have reached here. Again, we have run a query tabular. So we are now using the concatenated data sets, and then we are running the query. So this will give an SQLite file. Now, so till then, I think we have completed the first part of the workflow. So here you can just go through this workflow that is already given by Galaxy. We can go through it and understand it, because it takes a little bit of time to understand what we are doing until we get some result, until we get the final result, of course. So what all each step does and what we have to do to reach up to that level is exactly to be understood by you when you are being in a workshop. Right now I'm not able to see any faces, so I'm not able to get whether you are doing it, whether you're doing it right or not. So just feel free to ask any questions, and I will share this tutorial link so that you can just compare the results with what you have obtained along with what is there in the tutorial, because the tutorial actually uses some of the older versions of the tools. So running the tutorials might not always feel the same as that is there. So a few changes have been there since the tutorial has been made. I think some tools are updated. So I'll just share the link in the chat box. So the participants can take 1 or 2 minutes to just go back and see how the tutorial works. You can just go and have a look and then come back in 2 minutes. That would be fine, ma'am.
Yes, that's perfect. So now let's wait, and then maybe you
Yes,
can
yes,
provide an overview again of what you
yes, yes,
finished in workflow 1, and then we can move to workflow 2.
yes, ma'am. Other than custom ProDB, is there any other R package in Galaxy platform that can be used for genome mapping? I'm actually quite not aware of that, but I think custom ProDB matches all the requirements. Then I don't think there is any other method that is more suitable, because it almost integrates everything that would need for the further analysis. So I would recommend custom ProDB. So if there is any other tool that can be used, I will just check and let you know. I think a jitter channel is working for replying to the queries, isn't it?
Yes, it is working.
Yeah, you can just paste the question there and I will get back to you.
Yes, it's always useful in Gitter channel. You can paste the questions, and anytime the speakers can come back, not just today and some other day also. That's possible.
Any questions from the audience?
I don't see anything here on the chat, neither on the YouTube channel. So
I think since it is the first time they are doing this workflow and it's a little bit complicated to understand why we have converted the file into different formats and finally we are recombining them. It's a little bit tough for the first timers.
yeah.
It's
Yeah, but
okay.
it's important that you are detailing the steps, and it's also important that you are making them understand the significance of when a program reads a file. It actually reads the content in a way where you actually the program is not really um written in a way that it can read the file contents. It won't run.
Yes, yes.
And this is why workflow systems come handy, because now you are not doing this all by yourself, which is one of the major limitations when you write your own workflows. That is why Galaxy makes it a lot easier. That is basically brings out the value in terms of how somebody else can just rerun this entire workflow without creating one.
Yes, yes, exactly.
Yeah.
I just want to say one more thing here. Um, right now, only one registered user is doing so the Galaxy thing. I would encourage others also. Please do go to the usegalaxy.in, or let's say usegalaxy.eu, and then you can do the workflow. The workflow link is already provided. And yeah, if you require any question, anything, please do ask in the chats or also the Q&A, whichever mode you'd like to.
Okay.
So, Sona, just one clarification. Once you are done with the workflows, are you planning to uh, have time for the participants to do hands on right now, or you would recommend to do it later after the session is done?
Ma'am, I think it will take around 45 minutes more for completing the entire workflow. So that is why I wanted to go with this explaining the history based on the history that's already created. So the participants can either they can just go and do it later and then come back, or they can do it from the first workflow. They can maybe they can run it. No issues in that.
All right. Okay. So, as you know, Sona is saying that she will actually give you time, so that if you run and do an initial exercise right now, it'll allow all of you to understand what challenges you are facing. She's right here, so she can solve some of your queries right away. If not, and if you plan to do the workflow later on, then of course we are available to help you out.
Yes, you can paste the questions in Jitter. I'll be there always. So you can I'll be happy to take all your questions, but I think a little bit it's the topic itself is a little bit tough for beginners. So you can just listen, you can understand what each step is doing. Then when you finally have your own data, or if you want to analyze some data, you can always come back. I think it is you streamed in YouTube, right? So you can always view and then check it out.
Right.
Okay.
So I think let's proceed to your second workflow.
Yes, yes, ma'am. So again, we will just come back to what we have done in the first workflow. So if you remember all these steps, they have been done in the first workflow. So what the difference is like when we are seeing this workflow, we'll be feeling that it is a very small workflow. But right now, I think you must have understood how many small steps are there in between each of the tools that we are using, how each tool accepts the input. So all these things are there. So right now, we have just made a database from the first workflow. So next, in the next workflow, I'll be telling you how we are going to use the database, and then we are further analyzing the input sequence. So now in the first workflow, we have just used the RNA-seq FASTQ files and the GTF files. Now in the next workflow, we will be needing the mass spec files. So we will be going on to that. So we have in this for this proteogenomics tutorial 2, we have some files, some mass spec files. So they are basically MGF files or the mascot generic format files. So you can download or you can just copy. If you're just uploading it, you can just copy and paste it and upload it. And you can just convert it into a list, and you can save it like this. MGF, it is a list with 4 items. So these 4 files, or the MGF files, are the inputs of our proteomics part. Now, in the next step, similar to what we have previously done, but I think we'll just be introduced to a few new tools, one of which is Search GUI. So in Search GUI, you have the option to give the parameters of the proteins, how you have to digest it, all the information um, regarding the proteins and the precursors. All these information can be given, and the Search GUI will create a custom database that is covering all the information that you have given here. So I think Search GUI has a new updated version of the tool. So I think Search GUI will not be performing well on the workflow 2 that is already there. So I think in this Search GUI, now you will have to give the parameter file as a separate one. Let me see, I'll show you. So here you have to, unlike the older tool which we have been using in the history, we have to create the parameter file and we have to give it here. So the parameter files can be created using the identification parameters, and here you can just give the details regarding the modifications, variable modifications, and the digestion details. And that is a separate file. I'm talking about the updated version of the tools. And again, you have to run Search GUI. But for the time being, since our history is based on the previous one, we can directly see what is happening here. So similarly, it has taken the FASTA file and input the peak list. We have a collection of 4 spectral files, the MGF files. This is a collection. It is inputted as a collection. And the database search engine is Extendum. So all these parameters you can just give as an option here, and you can just click on execute. So it is an archive, so we cannot view it right here. So now the next tool is the peptide shaker. So peptide shaker will give us the peptide spectrum matrix. So we will see how to run it. So here we are taking the input of Search GUI, and we are just directly running. So we can create a mzident.ml file, and we need not include the protein sequences. Then all these things can be avoided. Then we need a peptide report, a protein report, and a certificate of analysis. So all these can be checked, and you can just click on execute, and you will be getting so many files here. We'll see each of the files and see what it is saying. So we have the parameters, then the PSM report, then the peptide report. So all these files are here. Now, this is one important step and one important file. So we are taking in the output from the peptide shaker to create an MZ to SQLite file. So here, earlier in the previous step, we have created an MZ-ident.ml file. So we are using that as a proteomic identification file, and then we are giving the spectrum, and we need the database. So this is the purpose of the database which we have created in the first workflow. So using this database, we are searching, and we got an mz.sqlite file. So we will see the contents of this file later on in the workshop using the MVP viewer. Now, removing reference proteins. So now it is time to filter out what we have based on the spectrum. So here we have taken the PSM report or the peptide spectrometrics report, and the database table is the peptide shaker, and we are giving the tabular data set, that is the reference protein accessions. So these have been created in the previous workflow. So what is our query? So we are going to give an SQL query here, and in this query we are finally ordering by the PSM IDs. So finally we will see how the file looks like. So now from a huge list of proteins, we have a few proteins based upon the SQL query which we have given. Now we have to keep another filter, that is selecting the sequence with length with greater than 6 and less than 30. So that is the next filter which we have given using the query tabular again. So this query tabular, you can see how useful tool it is while querying the database, or for any other purpose also query tabular is very good. So we are, you might be aware of applying the filter option in the Excel sheet. So query tabular is just like that. You can just query your input files in whichever way you want. Now here the input is removing reference proteins, the previous one, this file, and then you can see the options. So what are the column names that are to be specified? The ID, the protein, and the sequence. Then the filter data set input. All these information can be given. And here you have the SQL query. So here we are filtering based on the length. The length of the sequence should be greater than 6, and it should be less than 30. It has to be grouped by the sequence. So we will see the result. So now we have 6 lines of sequences, while in the previous step, how many did we have? We had 9. So 3 have been filtered out based on this criteria. Now we have to create a set of peptides for the BLAST P analysis. So with that, we have completed the second part of the workflow. That is, we have created the RNA-seq database, and along with the MGF files, we have performed search GUI. Then we have given it as an input to the peptide shaker, and finally we got an MZ to SQLite file, and we have peptides for BLAST P. So until then, you can just, if you want, you can just again go back, or I will just show you. I'll just give you the link to the second workflow, second tutorial. You can just go through that and see what happened in the second part of the workflow. I'll give you 2 minutes time. So the tutorial here also mentions the same things, but I think what is different from the tutorial and what we have done is like the way we are giving input to search GUI is a little bit different. So in search GUI, if you are ever doing this or if you are trying to do this today or tomorrow itself, you can just remember the fact that search GUI will require 2 files, the parameter file and then the main GUI tool, search GUI tool, in which you have to input the parameter files separately. So this is the data upload, then matching the peptide sequences, then here you have the search GUI. Whatever I have mentioned in the while explaining the workflow, it is all here. Then the modifications, the fixed and the variable ones, and the database search. Then you have the peptide shaker. and the certificate of analysis. So that is why we are getting so many files associated with peptide shaker. So we are in this step, we have created a file that is suitable for multi-omics visualization platform, or the MVP. And next you are doing the query tabular, in which we have extracted specific names, column names. So all these queries and all these regexes actually are a part of making the file in the way which you want to use it when you're running any tools of importance. So mainly you just have to concentrate on the major tools, and the rest, the file formats and everything, you can just do it based on your logic. Actually, the workflow will incorporate all of these steps. But the thing is, if you are creating a new workflow of your own, or if you are using your own data, then you will have to modify the workflows a little bit, so that it can accommodate your information also. And most of the time we will have to change the parameters when we are doing some workflows. So in some case, in most of the time, usually the defaults do not work. So you will have to have a small idea about what each and every tool does, so that you can modify the parameters without having problems in the further steps. And this SQL query is a great functionality, which I would say, in Galaxy, because it enables searching for things which it is present in the your big query, in your big files, and you can immediately get the results and convert it into a suitable file. So imagine what all you will have to do if you are using it in an Excel file. You will have to save it, you will have to again upload it. So it can be easily simply done using Galaxy. So these are the so now we have a set of protein sequence that are ready for blast. So this is actually the contents of the proteogenomics 2 workflow. You can just go back to this if you're when you're doing it. So now again we will just come back to our last part of the workflow. So now we got the blast P results. Now we need to find out the novel peptides, if there are any. So we will see how it can be done. So we are running again query tabular on the peptides that we have got, and again the tabular data set for the table.
So now question is there.
Okay,
Mm,
let's just come back to the chart
yeah.
with the details. So can the
I can
second,
read you. Yeah.
yeah, no,
Okay, go on.
I'll just go on. Can the second workflow started directly without doing the first with the data? And same question for workflow 3, which you are going to start. Okay, so do you mean to ask, like, can we do the second workflow with the MGF files without having the raw FASTQ files, or is it like somebody has not started the workflow 1 and they want to go with the workflow 2? Is it like that?
Maybe cover the both the scenarios.
Okay, so it is actually not possible to go on to the second workflow without doing the first, because actually database is the heart of proteogenomics. So without a database, it is actually impossible to find out anything novel, or any information. If we are not taking into account the first workflow, we are not considering the genomics part, we are considering only the proteomics part. So if you want to do that, if you want to go on to the second workflow without doing the first workflow, you can always import the second workflow and then do the analysis using that. With the available history, you can always import a history from that is created by Galaxy P, and you can use that, and on that you can run the second workflow. If somebody wishes to do that now, they can always do that, no problem. But originally, if you are doing the experiment and you are getting the mass spec files and as well as the genomic files, where would you input the genomic files if you are not doing it in the first workflow? It is not possible. So, does the novelty of the peptide depend on the propensity of any single or group of amino acids? So, the novelty of the peptide, like we are giving an in silico candidate for validating the peptides. Actually, we are not, we are claiming that it is a novel peptide because it is not present in the databases, but still you have to validate it. And whether it is depending on the propensity of any single or group of amino acid has to be evaluated through the validation step only, because as far as databases are concerned, they will just return whatever that is not there in the database. So I hope that clears the question. Now,
Yes.
any other
Uh,
questions?
I just want to add to the question what the first
Yes,
one was
yes,
there. As you said, with the history, it's possible. Would you like to share the history, what you have right now? Is it possible for you? So
yes,
that they can
yes.
start from the third workflow if somebody want to start?
Yeah, yeah, sure, I'll do that.
Yeah. Maybe you
Yeah.
can actually try to show also, then people will know how to share the history.
Oh, okay, okay. Okay, so I didn't go into much detail because I thought it was covered in the first pre-conference meeting.
Yes.
So it's
Yes,
okay,
yes.
I'll just do that.
And some people are still new, so it would be good idea for them to know that.
Okay, okay, fine. I'll just show that. So whenever, if you feel like you want to do the analysis from the second workflow or the third workflow, so Galaxy has provided all the data, all the information. Like whenever they are conducting some tutorials, they'll be providing all the information in the histories and as workflows. So I'll just show you how to just go for that. So here you have, first I will just tell you how to import the workflow. So here you have in the shared data menu, you have the workflows. So a lot of workflows are there. You can just scroll down and see for different purposes. They have created so many workflows, and so many are run by participants like us, and we have shared the data, or they are, some of them are done by the Galaxy team itself, like by Melanie Fall. So if we want to import any data set, like for example, right now we are doing proteogenomics. I'll just go to proteogenomics, click on proteogenomics, and then you can see all the workflows that have been, that are coming under this name. So here you can see, just to make sure that it is authentic, you can just see that it is done by the Galaxy team itself. So these are all by the Galaxy P team. And if you want to click on, if you want to import a workflow, you can just click on and click import. Or if you want to run it on the spot itself, you have an option to run it. So I will just show you how to import it. You can just click on import. So here you have an option, start using the workflow or return to the previous page. I will just return to the previous page and just show you from where I can get the workflow. So now you have imported your workflow, but where is it? So you have to take it from the workflow menu here. Here you have the imported workflow. It has been updated a few seconds ago. That shows that you have recently downloaded that. So if you want to run the workflow, you can just click on the run workflow. So if you click on the run workflow, you can see here what all the workflow is taking in as input. So these are not the right input files, because in this in this workshop, I have created a few other files also, so they will be providing so many options. So if you are starting from scratch with the workflow one, and you have only three files like the reference, then the GTF files and the FastQ files, then definitely you will be able to see only those three files, and those will be coming here in the right way. But since we have so many files in our history, it won't be coming like that. Or another option is like you can import your workflow into a new history. I will be showing you that also. So if you are planning to use it, you can just click on run after selecting the right parameters. So the workflow includes all the steps that I have told. So from the starting to the end, all the tools that I have used, all the parameters that I have kept for each of the files to run, everything is there. You don't have to modify anything unless you are sure that your data needs to be modified according to some parameter, and it will give you the result within a few minutes time. So this is how an imported workflow runs. You can just click on run workflow, and it will be running. So it's always better to go use other history. So here you have an icon like this, view all histories option. You can just click on it, and you can just create a new history here. And in this new history, you can import the workflow. So currently that is my history. So you can just go back to the main page. Here you can use the imported workflow here, but it will not work here because we don't have the input files here. So if you want to input the files, you can just go to the first workshop material. You can just copy the location in Zenodo. Just click on upload data, paste and fetch data, paste it, then start uploading. So the three files are there. Now if you click on run workflow, I'll just wait for it to run and it's completed. So now we know that it is queued when it is showing gray color, and it is running when it is in the orange color, and once it has completed finished running, it will be shown in the green color. So now it is running. So when it comes to green, you can see that it will be loaded here itself. It's taking a little bit of time, but it's okay.
So a couple of more
Yes.
users are doing right now. So
Okay,
maybe
so
we
that's why, yeah. So you have the input. Now you can just refresh it, and see whatever files I have mentioned. Like earlier in the previous history, I had so many files, but right now the program does not have any other options to take other files, but it has to choose the right ones itself. So here we have the FastQ file, then you have the GTF file, then you have the reference, then you can just directly run the workflow by clicking on to run workflow. If there is some error or at some point you have forgot to mention some parameters, it will be asking in the workflow itself. So we will see. So I think we will just go to the next one until this one is finished. So anybody who has parallelly run the workflow, they will also get the results accordingly. So the here you can see that once the workflow is being run, all the files have been created. Whatever I have shown here, all the files have been created. So they are waiting for running. That's why it is shown in gray. So by that time it is finished, we will just go back to our previous history. You can always switch between the histories by clicking on to switch to. So we will continue with the last part. So till then I think the workflow will be completed. So here you have got the BLAST P results, then novel peptides. So based on the query, we had a few novel peptides. Now we have another option to see how the spectra looks and how what is different in the spectra and how we can compare the different spectra. For that we have a tool that is called MVP. So for accessing MVP, so we have to find the MZ to SQLite file. So that can be opened using the MVP viewer. Here you have the MZ to SQLite file. You can click on MZ to SQLite. Then click on this graphical kind of symbol, visualize this data. So here you have the MVP application. Click on that. So here you have the MVP viewer. You can also open it in a new window instead of in the Galaxy itself. So here this gives the list of peptides with their corresponding spectra counts. So here we don't want to know the information about all the peptides. We just want to know the information about the novel peptides. So in that case, you can just always click on the load from Galaxy. Then you can click on novel peptides and click on use for filtering. So if you click on this novel peptides, you will get the results of the novel peptides. So the total number of spectral counts will be 3 plus 1, 3 plus 3, that is 6. So this particular peptide has got 3 spectra, and we also have the protein count associated with that. Now I would like to view the PSMs for the selected peptides. So you can either select 1, 2, 3, 4, all the peptides, or you can just select it any one or two of your interest. So we can just select the PSMs for the selected peptides. So this will give you the result consisting of information like the spectrum ID, the spectrum title, then the comparative theoretical mass, then the PSM score, then the confidence value, the tandem expect value, then the type of confidence, then the YIN, BIN details. So all these things you'll be getting in this. Now, if you want to see the image of the spectra, so here you have, if you click on the sequence, here you have the Lorikit viewer. Here you have the details regarding the spectra. Here you can modify any of the details and see how it is affecting the spectra. So you can add the ions, and you can also label the peaks. So many information can be altered, and You can just see what the B ions and the Y ions are, have the values here, and the spectral information. So all the protein files, all the PSM files can be viewed like this. You click on this, and there is an option, like if you like a spectra, you can always just click on like, and if you don't like the spectra, you can just dislike it. So usually the spectra that has B and Y ion values of three continuous ion values will be good. So here I have three continuous values in the B ion, but in the Y I don't have any three continuous ion. So that option is to dislike it, because it is not the kind that we would want, but it depends on the user. Now similarly, we'll go to the other spectra. So here we are getting a continuous values for B and Y also, so we can just give it a thumbs up. Now we'll go on to the next one. Here also we are not getting three continuous B's and Y. We're getting three continuous B's, but we are not getting continuous Y's. So it is not good. Then here for this peptide, you have three spectra. The same peptide itself, you are having three spectra. You're having continuous B's and Y's. You can just click on like. Here also no continuous B's. So likewise, you can select your favorite spectra, and you can just view that and get the information from that. Now you also have other options, like you can have the information regarding the peptide and the protein, like in the whole protein length where the peptide aligns. You can get the information from here, peptide protein viewer. So in the selected four peptides, they are asking whether I want to view which one. So you can just click on that, and you can just click on here. Then if you just go above, it's not coming, I think. Okay, we'll just go on to that from here. So I forgot to tell one more thing. Actually, we have an option to export the scan values that we have obtained. We have selected two spectra, so we can export that to Galaxy. That option is also there in MVP viewer. Let's click on export scans. Now we'll just see the protein peptide viewer. I think it is not coming, protein peptide viewer. So there was an option to view the proteins and the peptides along with that, and that is here in the protein peptide viewer, but for some reason it is not coming. I'm not sure why. Okay, so here we imported the information from MVP and we have pasted it here. You can just view the file. Whatever information that we have got there about the spectra of our interest are imported to Galaxy. Now we stopped with this. We got the gene, like we are interested in these two, so we will be running the genomic coordinates on this data. And here you can see the peptide genomic coordinate tool will give a result like this, consisting of the information regarding the chromosome number, the chromosome start and stop site, the name of the peptide, then the score and the strandedness, all these details. Now we will just go on to pep pointer. So Pep pointer is a tool that can classify the genomic location of the peptides. So it will compare the original reference genome to that of the peptide genome coordinates of the peptides of the spectra in which I am interested in. So this gives the pep pointer data. So from where it is located in chromosome 2, the name, then the score, the strandedness, and also it gives a small annotation regarding that. So it shows that overlapping on two regions. Please look manually. So we have to look manually means we have to search the MVP tool for that. So as a final summary, we will see the details about the novel peptides. So similarly, here also for getting the final summary, we have done the Curie tabular on pep pointer and the PSM novel peptides. And here you can see the results. This is the final summary of the peptides. Here in the last line, you also have information regarding the UCSC genome browser. Just click on copy this and you can just paste it here. So you are redirected to the UCSC genome browser in which you can get the location of the peptides. So now I think we have come to the end of the tutorial, but it is always good to just go to the MVP viewer and get additional information. So you can do so many things using the MVP viewer. You can just filter using the scores and you have so many information from here. So I think how many people might be doing this workflow, Dr. Kiran?
I can check just a moment.
Yeah.
Looks like a bit increased now, but
Okay.
so there are some jobs are running, and yeah, I think
Yeah.
at least 8-9 users are doing it, or maybe more than that.
Okay, great. So I think okay. Any questions? No questions. So that is actually the stepwise, extraction of entire anatomy of the workflows. So when you are doing it, you will be most probably requiring the information regarding each tool and what input it takes and how it is running. So as of now, if you are comfortable in using the workflows, you can just use the workflow as I have previously showed. So and another option is like you can use the Galaxy tutorials and they will be updated with every Galaxy smorgasbord sessions. So you can just use that and use your data like that. So if you are if you are not using the workflow also, you can just directly copy and paste each line from the proteogenomics tutorial. So that will first I would suggest that you should use the tutorial itself, because instead of running a workflow in which you are not aware of what is happening behind, it's always good to run the this thing run the tutorial step by steps and understand what the parameter stands for, what each line or what each tool does and what you want out of it. So I think that is the best way to learn from scratch. uh, so with that, I think I will conclude this workshop. The users can do the workflows now itself. I think we have time, isn't it, ma'am? We have.
Yes, definitely we have time. So there are a few questions that I saw earlier. I think you answered most of them, so we are good.
I'm curious to know what is the length of the peptide in general. So based on the cutoff, we'll be keeping the length of the peptide like less than 30 and greater than 6. So that is what we have kept in the Galaxy. This one, the workflows. So it depends.
And for general information of all the participants, see, anytime you do any data analysis, obviously there is a question that you are trying to solve, right? There are several publications where Galaxy is used for proteogenomics to study novel protein variations or to look at mechanisms of mammalian hibernation via quantitative proteogenomics. So there are several very interesting questions that one can ask. The advantage is, since a lot of this data is available in public domain, now you can think of a completely new workflow system that a new question that has never been asked before, and use the tools that are provided in Galaxy as a workflow system to really answer those questions now. So it's not just specific to uh, a very tight made question. You actually now can be more creative in looking at what kind of data sets are available in the public domain, what you can use, and how you can use those data sets to answer new questions. So that's pretty interesting, because it's like, I see it like as playing a game, because then you have some data sets and then you are trying to play with it to see what answers it is giving you in terms of, can you identify novel proteins in different contexts. The data sets, good part is a lot of publicly available data set does exist, so try that.
Just like ma'am said, actually we have a rich amount of information from Galaxy themselves. They have created so many tutorials, so many workflows. The only thing is that you have to spend a little bit of your time. You have to invest in that. You can find out what is happening in Galaxy. Not only Galaxy, even if you consider programming also. In if you take our statistical package, you can see that every day it is being updated. So all the tools that are there in R, I think Galaxy is trying to incorporate them here also, so that the non-programmers can use them. So it's like, take this opportunity, like whatever data is there, you can just download them from NCBI, SRA, then from the Pride database. You can just try to combine them and see if there is something novel, something new. Like, you have everything with you. The only thing is that you have to spend your time on that, and you have to read a lot about it. But I think it is very much worth it, because I have been using Galaxy for the past 4 or 5 years. And initially, before using Galaxy, I was not having much programming knowledge. But after running the workflows and after using the steps in Galaxy, I could really do it on other programming languages like R and Python. So it was a very user friendly workflow, which actually could dive deep into the exact mechanism of what is happening in that.
So I don't see any questions on YouTube or in the chat window, but I think Sona, you did a really good job
Thank
at explaining the workflows and going through each step in Galaxy in a detailed fashion, so that there is clarity on the input file formats, the output file formats, why the formats are important to invoke certain programs, what kind of changes one can make using regular expressions in order to bring about a file wide change. So, and then how data sets generated at different steps in the workflow can converge in the towards the end of the workflow to answer questions. So that kind of a thought process, that kind of data analysis understanding can come only when you practice.
Yes.
So that is, I think Sona practiced a lot, and that you could very easily see when she was explaining it with the level of clarity that she has. So strong recommendation to all the participants is, keep doing it over and over again, so that you understand the steps. The more you do it, the more clarity you have on what is the significance of individual steps, and that will allow you to think more clearly in terms of what was really happening with the data, right? So
Yes.
Sona, if you would like to add something, and then we can hand over it to Kiran.
Yes. So if any of the students are interested, they can directly contact me or Anshu ma'am for any queries, because we are constantly using Galaxy, and we are just keeping ourselves updated in this regard. So if there are any queries, I would recommend you to clarify it with any of us. And there are a lot of experts here also. Kiran sir also can help you. So I think you have almost all the resources that are needed to get started with what you are doing. So and the best part is like everyone requires an online free tool for doing their work. So Galaxy is the best platform, I would say, in that sense. And people who don't have programming experience are also can just do their RNA-seq analysis or whatever analysis they have with them, because they have a number of tools, I would say, to clarify everything, all the data generated, everything that can be done using in Galaxy. So you can just give it a try. And anytime you can just message me. I have shared my email in the beginning slide, so you can just contact me. So uh, that would be my advice to the youngsters, just to keep on practicing, because at one point of time we all were beginners. So it's like how much we practice, how much time we put into it, and how much we, I mean, when I had doubts regarding Galaxy, I used to message to Dr. Dave Clements. I'll just clarify things with him, because that is the only way. Initially, that was the only way to clarify things. But now we have so many forums, and we have so many bioclose users also. So when you have a question, you just have to ask, you just have to shoot it like that. You don't think that it is wrong, or it's going to be a blunder, because everyone does that. So that is how we are learning. So that would be my advice to the students.
So that's a really wonderful advice, Sona, because I you really captured the essence. We all start learning ABCD. So yeah, so that's the essence. So if you're interested, just get started, and whenever you are stuck, there are so many of us to really help you out. And with that, I think we should really applaud Sona's effort in actually giving a great demo and a very nice session. So congratulations, Sona, for that. Very good. And I would like to now hand over the session back to Kiran.
Great. Thank you, Anshu. Thank you, Sona. It was really wonderful. I would say it was an extensive step by step, each procedure, everything. Um, I mean, let's say a bit sadly initially that not many started, but later they took up and then picked up. That makes they were happy to do that. So that's really good. At least some people I see now it's raised to 16 members are doing it. So hopefully they are still executing. Keep doing that. And so while you do, we want to see your bright faces and smile faces so that I can take a picture. Keep doing and then show your bright faces with that. And rightly said by both the people, by the both Anshu and Sona, it's like just invest the time, and we want to see all your errors, not the success stories, but all your errors. It's not the success things. Success things you can keep with you. It's fine. So on a count of three, I take a picture. Three, two, one. Thank you. So yeah, that's all. And of course, another exciting session is going to come, and the next speaker is already available. But we start at 3:30 as decided. If some people are left and they want to come back, so please come back again at 3:30. The session will be open here, and you can always go back and then come leave and come back. But we'll be open. Keep open this one.
I think I'll be available till 3:30, so that any participants having questions can ask till then.
Yeah, great. That's great. Yeah, they are executing, so maybe that's
Yeah,
definitely
yeah, I
useful
think
if
I
they
should
are asking.
be.
Sure. So, in another 2 minutes, we will start the session. Stefan, can you hear us?
I think so. Can you hear me?
Yes, but it's a bit echo
Wait,
from. Yeah.
just a second.
Mhm.
Is it better, Kieran?
Yes, a bit better, but it's still a bit of thing. Maybe can you say something again?
1, 2, 3, 4, 5.
Yeah, it's better. Yeah,
I think it was the second microphone.
yeah. So wonderful. Welcome all. As being myself, the chair. So, okay. Thank you, Stefan, for coming to this session. Although you had some accident issue and other things, and you made it here. Thank you very much. I hope you're doing fine. And yeah, about Stefan. So Stefan actually he studied his bioinformatics in Berlin Free University, and then he moved to Freiburg to pursue his PhD in the University of Freiburg in the Department of Pharmaceutical Bioinformatics. He has done extensive work in various publications of epigenetics area during his PhD. A lot of collaborations he had in this epigenetics area, and later he has done his postdoc while contributing to the Freiburg Galaxy team. And there he has a lot of exposure to the galaxy. And before earlier, also he was doing a lot of galaxy works. And presently, he is a freelancer at Grunenthal for machine learning based drug design projects. So, Stefan, it's all yours. You can start, you can share, and then you can start your session.
I think you need to stop your sharing, huh?
Yes. But yeah, multiple sharing is possible, but yeah, I stopped sharing.
Just a second. I think it's this one. Is it there?
Not yet.
Why not?
You're able to share, or is it like giving some issue? Okay, just let me check.
I can stop again and start it again. Wait.
Yeah.
Zack. Zack. And I shared. Okay. Then we
Yes.
try. Is there?
It's there, but it's still a black screen. There's nothing in that.
Okay. And I'll share another one. Just a second.
Sure, take your time.
Does this work now? Still black. I'm not sure what to do though. Should I unblack myself or something? Because it's so. What do you propose?
Do you have multiple monitors?
Yes,
Okay, and is it Ubuntu? Or
it's Ubuntu. Yeah.
okay, think so. You should be check if your two monitors displays are visible for you in the display settings.
So I click the window so that I want to share the window, not the whole monitor.
Okay.
Um,
Maybe try to share the monitor if that's a possibility.
so stop. Maybe I can. I don't know. Should I try Firefox?
Yep, go on. That should be fine. Come back again. I'll make yours.
I think.
We're still waiting, trying to get Stefan. We'll take another couple of minutes more. There's some problem. Okay. Great. So Stefan is back and is able to share. Okay, but it's not on the share right now. Did you stop?
It should be there. Yes.
Now it's coming. Yes. Okay, great.
Let me start my windows again. 1 second.
So while he's doing his login into Galaxy, all the participants also can do that to the Use Galaxy. It will be another engaging probably because you will also can do simultaneously, most likely. It's a workshop.
I'm done in a second. So now everything's with it. Yeah. It's a bit slow though, but should work. Yeah, so then let's start.
Great, thanks. Go on.
Okay, perfect. So as Kieran has already mentioned, this is also thought to be some interactive session. So please feel free to log into a Galaxy instance of your choice. The EU worked fine, but yeah. Please remember that you start with the new history. You can also use an old one, but yeah, sometimes the participants get confused when other material is stored in history. This session will give a rough introduction about the possibilities of epigenetic analysis in Galaxy. And when you go to the Galaxy Training Network website, there's a subpoint epigenetics. And when you click on it, you will find a couple of very nice tutorials. I decided not to start with the complete introduction because there are some slides available and also a recording of this introduction. So maybe it's a good idea to anyway have a look at it. The basic principle of epigenetics is explained in the introductory part, but I guess there was already a short session yesterday, I think, that gave you at least a basic idea of what's possible. I will mention probably some keywords that are maybe not clear, but feel free to ask in the chat if something is, if that's the case, and I will try to answer it. And don't worry, everything is new. Just try to follow, get a first impression if and how the stuff works, and then it's fine. The tutorials that are available can be clustered in a ChIP-seq part, and there's also a microarray based DNA methylation analysis available from Illumina. Maybe array microarrays is more a topic in some labs. So yeah, and we will today work on the ATAC-seq data analysis. And for this, there are also some slides available. It's not really required to have a look at it. We will directly go into the hands-on. Yeah. Requirements for this sessions are a rough idea how quality control works with epigenetic, microarray, or NGS data sets. If that's not clear, it's not a problem. I will then just probably use words you don't know. Then have a look at the slides or even the handout of the quality control tutorial. And we will do some mapping because we are dealing with reads of an NGS experiments and we need to align them to a reference genome. How that in detail works is explained in this tutorial. It's also not important that you don't know it in details, but if you're more interested to go deeper in that topic, just have a look at this. Yeah, today, as I mentioned, the idea or the story of this tutorial is the ATAC-seq dataset or technique. I will soon explain what it is. And we will do a basic pre-processing of public available datasets. We will do the mapping part to have a rough idea of what we measured. And then we'll do the most interesting part, probably some small visualization steps. Exactly. To give you a rough intro, in this training we will learn the general principles of this ATAC-seq data analysis. This technique is a method to investigate the chromatin accessibility. Therefore, the genome is treated with some transposase enzyme that is called TN5. This is a word we will from time to time hear today. And these transposase enzymes mark specific part of the chromatin that are open, so not that are not connected with the histone. To have a rough idea how that looks like. So just as a reminder, you have the DNA, and these DNA fragments are somehow bind to histone fragments. And what are we interested in today is the other small DNA fragments in between two nucleosomes. So we call this the open chromatin area. The training material will give you a rough insight on how to quality control the data. You will look for, so this is an idea of the workflow today. You will do a small quality control of the data. You will look here for low quality bases, adapter contamination. You will check the correct insert size and if there are PCR duplicates available. This is stuff I will explain later. We will see how to remove these datasets with a tool called CutApt. We will use FastQC for quality control and some basic plottings that give you a rough idea if everything works fine. We will map the reads with bowtie, and afterwards do some also pre-processing steps. Then we will use MAX2 to identify chromatin regions that are enriched in our measurement, to identify peaks that are of interest. And we will use PyGenome tracks and deep tools to get some basic idea about possible interpretations of the dataset we used. Therefore, we will have a closer look on a binding protein called CTCF. I will also explain this later, and yeah, then have an idea what we measured and if everything somehow gives a conclusion at the end. So the topic is, what is ATAC-seq? What are probably quality parameters, good quality parameters that we should use, and we will learn how to analyze the data set, and we will hopefully understand it, and the peak calling steps of which regions are enriched is the most important point, and hopefully a rough idea of this is clear at the end. To go back to this screen, in many eukaryotic organisms, like humans, the genome is tightly packed and organized around these nucleosomes. One loop around the nucleosome is roughly 150 base pairs long. This is a number we have to keep in mind because we'll see it later in one of the histograms. And as long as the DNA is packed around these nucleosomes, it's not really possible for the to copy the underlying DNA to RNA. So it's very important that this area is open, because that somehow leads to the possible usage of the gene. Yeah, that's why we want to analyze this area. And many factors, such as specific chromatin structure types, the position of the nucleosome on the DNA, some histone modifications that are also shown here that somehow have an effect on the packing and the opening of the histones, play an important role in the organization of this DNA and therefore also for the accessibility. And this is something we want to check today. Which of these factors are present, which are not present, which play an important role. And yeah, for this you can use this ATAC-seq. Exactly, we can go into the tutorial. The ATAC-seq is a short term for assay for transposable accessible chromatin using sequencing. And the idea behind this is that you have small GN5 transposons. They are mentioned here or shown here in red and green, that bind to these open chromatin signs in between, and then you can afterwards analyze the presence or absence of these small elements and therefore identify areas that are open. There are more techniques available, for example, FAIR-seq and DNA-seq. I will have explained that today. The ATAC-seq is somehow the most popular at the moment to deal with this data set. Exactly. Another term that is important today and will drop a couple of times is a transcription factor or transcription start site. That's somehow the beginning where the transcription of a gene starts. And there's a very there's an area where a lot of regulatory elements can bind, and therefore, if this area somehow is open and not packed around the histone, you can somehow have a closer look at it. And this is important today. You should know what a promoter is, because it's not only the transcription subtype, but also the area before. And yeah, that's basically it. So then we can go into the Galaxy session. The public data set we will use today is stored on Zenodo, and you can easily import it. You can just copy it, but as a hint, you can also go to the see Galaxy training materials icon on the top of the screen and go to the epigenetics part, go to the hands-on of the ATAC-seq data analyzers, and compare to the normal visiting of the website, buttons like the UCSC table browser tool icon are then clickable, just as a hint. You'll see what it means later on. Copy the URL, upload the data, click on paste fetch, let's start. And please feel free to follow the steps. It's not that difficult today. I have to mention that probably I will skip some steps of the tutorial because the calculation sometimes takes longer, but I will have the resulting files there and will explain it. So don't get confused that I don't click the calculate button, but I will mention it then. The dataset itself is taken from a public available dataset. The corresponding paper is referenced in the introductory part, and will mainly deal with this DNA binding protein CTCF. There are hundreds of others available. This is just something we know, or we have pre-knowledge that this binding protein gives good results. Yeah. But you can, of course, also analyze other data sets with it. So the data set is there. Probably is also already mentioned that it's always a good idea to work with text. So just open it, click on the data tag, the BED file of this CTCF enriched regions should, I name it like this, CTCF. You can name it as you like, it's just for better overview. And the second also. So I called them R1 because it's we deal today with paired end data. So each read is somehow analyzed from both sides. So R1 is, let's say, the left side to the right side, and then they are connected in the middle. Sometimes they're also called forward and backward, or no, forward and reverse. Here they are called R1 and R2. And then the next step, we have to change the data type of the bed file because it's called interval. Let's rename it as bed. For the other ones, I think it's already correct. Yeah, fast using is fine. Exactly what a fastq file is and what a bed file is. There are also tutorials mentioned in the other tutorials mentioned in the ATAC-seq tutorial. Just click on the link and you get a more detailed overview. fastq is basically a faster file with more quality information, and the bed file is somehow stores way more information in the proper overview you get on this link. So then we need an annotation file because we want to map our reads against the reference genome. For this, we use the UCSC main table browser tool. And when you are in another tab, you cannot click it. But when you go in the Galaxy tab, click on training material, then it's directly here. You need to execute it, and then we are forwarded to another website. We are interested in mammals and human chromosome. We take this version of the from December 2013. Genes and gene predictions is something we are interested in. We need to take here an old version of the GenCode reference dataset. So 37 is important. Click on basic. We are only interested today in chromosome 22. You can also do the analysis with other chromosomes, of course. It's just that we keep our dataset small. We want output format all fields, and we want to send the output to Galaxy. And I think that basically send query to Galaxy, and then it should be there. Can take this chromosome 22. The format that the UCSC main table browser tool differs a bit from the one we require later. So we take a cut columns from table tool, and then we apply these parameters here. For better practice of following the tutorial and the analysis, it's always a good idea to have two windows open. So one that shows you the hands-out or the hands-on, and the second one, the Galaxy interface, that you easily can copy the parameters. So we C stands for column. So we take out specific columns in a specific order. We want the delimitation by tab, and we take our dataset that was just downloaded from the UCSC table browser, click on execute, and sometimes the tags are not shown. It helps when you refresh the history, and they are, and it will pop up. and then also the text are shown. You can look at this file and you see compared to the other ones that we got rid of specific columns and ordered them. So chromosome, start, end. This is probably an identifier of the genome, which strand. Yeah, more is not required. Exactly. So let's suppose that we rename the data set. It's not, it also works without renaming, but for a better overview, just name it and also change the data type to bet. And compared to the display before, because bet files have a specific format, each column has defined names. So like chromosome, start, and name, some kind of score, and the strand. Some columns are not present in our dataset, but it shouldn't be a problem for us. Good. Yeah, exactly. A description of more possible column names, column contents you can find on the GeneCode website, and the file is usually called the GTF file. So now we come to the first interesting step after finishing the pre-processing part, quality control. Because our data set is raw, we didn't have any let's say pre-processing steps in between, and you should always have a look on the quality part. And this is quite easy with Galaxy. You take for this the FastQC tool, and the parameters are quite simple. You just pick your two files and click on run. You can also do the quality control part on each file separately, but in our case, it's okay to click on the multiple dataset part, click both of them, and then click on execute. The other parameters can just stay default. So FastQC puts out two result files. First, a web page and the raw data. The raw data is a text file that you can use for plotting by your own. The web page part is way more comfortable to look at. You have a summary of different quality control parameters. Again, it's not the topic of this session to explain this in detail. There is one, it's just called quality control. Have a look at it. And there's directly explained which graphic, which parameter output has an influence on which graphic, and what are probably the most important points we should be concerned about. In theory, green is fine, red is bad, so nothing obvious here. What we will realize here is when you click on the adapter content part, we have an increase in the Nextera transpose sequencing read type fragments. And this is something we don't want, because the adapters are part of the ATEX-SEC tool or technique, but they have probably a bias on our data set. And we want to get rid of it because they actually don't mean something. They are part of the technique. The idea is that DNA fragments of about 40 base pairs are the results of two adjacent TN5 transposases cut the DNA in the specific area. And this can be smaller than the sequencing length of that area. And so we need to get rid of these reads. And this is the next step. So this should be removed. To deal with it, we need to trim them. And a detailed explanation how it works is maybe too detailed, but just keep in mind the adapters are not something we measured. The adapters are part of the technique, and we don't want them. For this, we use the cut adapt tool again. And apply these parameters. So our two read files can be specifically choose the adapter sequences we want to get rid of, and they slide the reverse and the forward type of this Nextera adapters slightly differ. That's why we have to apply them both. We also want to have a minimum length of 20, and we want to remove short reads also. What is important for us here also is that in theory we can use the cut adapt step also for both read files separately, but that can lead to the effect that one file in the end contains more reads than the other file. So we do it in one step that they somehow get synchronized or stay synchronized, because otherwise it can be problematic at the end. We pick the paired end, click the first one and the second one. With one option, insert 3 end adapters, enter custom sequence. We can name it, and we have to choose the sequence. The rest stays like this. And then we have the insert 3 end adapters. Again, enter custom sequence. to the filter options area. We want a minimum length of 20. Remove read modifications options. We want to remove short reads that are of low quality, and we want report. I think it is. Always keep in mind when you click and or before running a tool, take care that you really set all parameters, because sometimes they have a big impact and the result is probably empty, the tool fails, or yeah, always have a second look on it. Shabak. This can probably take a while.
So, Stefan, I don't see. I see, of course, at least two or three people are doing, but not many. So I would like to keep a poll. If people are given the history till here, would they want to carry from
Oh
here?
yeah, sure. I can share it.
Yeah, I will just keep a poll and then ask them.
I can also name it. I think it's this, right?
Yes. So participants, I just kept a poll. Please answer. Would be great to know. He can actually share the history till here, and then you can start doing from there if you want to.
I sent the link in the chat. So
Okay, great. So with the history, you can import the history and then start doing from there if you have missed anything.
so we can have a look on the report with a lot of data. The stuff we are interested in that area. Pairs that were too short, below 1% is fine. Pairs that pass the whole pre-processing step are nearly 99%, also fine. And now we check the removal part if these adapter sequences are gone. And you open it again, the FastQC tool. You click multiple files, you take the output of our previous run. So then we check the web page of one of the reads again, go down to our overrepresented sequences, there are none, and also the Nextera transpose sequences are gone. And this is exactly what we wanted. And to have a comparison with the one before. Let's take this one again. This was before. There are overrepresented sequences and next row there. No, they're gone. So perfect. Everything worked fine up to now. So so what we can also check if both files have the same amount of total sequences. Here we have 155 at the end, and then the second one hopefully also perfect. This is also fine. So when we go back to the scheme, we are done with the first part of it. So we imported our dataset, did some pre-processing, get rid of the Nextera reads, and now comes the fun part, the mapping. Because we have all these 280,000 reads, and we actually want to map them against a reference genome. For this Day-2 can be used. There are plenty of more available, and this is the one that is relevant for us. We use some pre-knowledge here because we extend the maximum fragment size of the parameters we want, or of the reads we want to map to 1,000, because we already know that this gives nice results. We will use the very sensitive mode to increase the chance for best matches, but also increase the runtime, which is not that important for us. We use the end-to-end mode because of the trimming step before. We can expect that the whole read of the map is available, and there is no additional clipping needed because we got rid of all these adapted sequences that are maybe influencing that. We use the reference genome that we previously also downloaded. The problem is here that this usually contains alternative loci. So some regions of the genome are present in both the canonical chromosome and on its alternate loci or loci. Our reads would then map twice, and this is something we don't want because it's very difficult to interpret at the end. And to avoid this, we use the canonical version of this chromosome reference, and we want to use. Yeah, sorry.
Stefan, there is a question. Sandeep, you can unmute yourself if you want to talk. Go on, Sandeep. Or you want to write, Sandeep, or you want to ask?
You can also write.
Yeah, he raised. You should be able to unmute yourself, Sandeep, if you want to talk. Okay, looks like he's not there.
Yeah, it's no problem. I can write and everyone.
Okay.
And another point that we will use is dovetailing. before AttackSec. I also never heard of that name. The problem is that adapters by this cut adapt step we applied before only get removed when they have a minimum match by 3 base pairs. And in our case, it can happen that there are only 1 or 2 base pairs available. And in that case, bow tie wouldn't recognize these sequences as matching or recognize them as non-cordant alignments and remove this. That's why, this dovetailing or dovetailing mode is available. And that's basically it. It takes this specific event into account. So that these mate pairs are still, or can be still part of the analysis. So these are the parameters we have to apply. We click on that, go to the bow tie. We are dealing with paired end data. We take our cut adapt output as an input. Do you want to set paired end options? Of course, we are dealing with it. We set the maximum fragment length to 1000. As I mentioned before, we use some pre-knowledge here. This is probably most of the time a question of experience. And we want to allow mate dovetailing. Then we'll just select the reference genome, use a built-in genome index, and set group information, do not set, and the reference genome, as I said, and that's really important. You have to take the canonical version, not the full. And we take the very sensitive mode. Can be that the calculation takes longer, but in our case it shouldn't be a problem because the data set is quite small. And we take the bow tie mapping statistics. That is not really required for the steps, but we can have a look at it. So very sensitive dovetailing. It looks good. Execute. Especially when you have a regular data set, the mapping part can take a while. In our case, it should be done soon. So we are currently in this step. We have the bow tie alignment step. We did again this quality measurement, and the next part will be to get rid of specific reads that are not of interest in our analysis. What that means comes in a couple of seconds.
So I just want to verify with the Sandeep. You still have the question? You can type in or you can also talk, Sandeep, if you want.
Perfect. Calculation is done. So we can look at the mapping stats and hopefully something is there. So 99 over the of the reads we added the bow tie algorithm have been aligned. That's perfect. If you remember the percentage before of reads that have been that passed the pre-processing step was 99.3. So yeah, some got kicked out. But this looks nice, and I think it's exactly also the stuff that's shown here. Perfect. Everything clicked correctly. So yeah, by adding the percentage of the read pairs, we come to a matching rate of nearly 98%. That's great. Good. So now we have our reads mapped to the whole genome of the human, and besides alternative load C on the 2 chromosomes, this was the canonical version, but there's still some reads in that data set that we are absolutely not interested in. First of all, we have usually a mitochondrial genome also present in the data set. And if we go back to the idea of the ATAC-SEC measurement, we want to measure areas between, or the accessibility of areas between 2 histones. Or let's say we want to identify areas of the DNA that are not packed around histones. Mitochondrial DNA doesn't have histones at all. So you have a couple of matches and alignments that you don't want, because there's absolutely no meaning in mapping, let's say, the DNA part that is anyway accessible. So the first part is getting rid of mitochondrial DNA. Again, we click on our training material icon, and we want to filter these reads out. Here we go. So we take our bow tie alignment. Don't worry that the colors of these tags is different from the ones in history. Doesn't have a meaning as long as the tag itself is the same. So this condition. Use the so we have a map quality, and in that case we can also use the larger than. So we are adding these parameters here. And the second one is proper pair. Proper pair because we have the pair end, and we only want to keep in mind keep the ones that are still there as a pair. Again, to get them synchronized, because when you kick out one, then the other one is useless. So we click it here, and this third one is the reference parts, and we don't want to have so exclamation mark the chromosome M, so the mitochondrial DNA. We don't want to set rules, we don't want a notification, that should be fine, larger than, as proper pair, chromosome. So let's see if that works. So the JSON part file we have here is not really interesting for us, but if you would run the tool in command line, you can apply a JSON parameter file. That's why it's shown here. For us, not very interesting. And then we have bump file of reduced size, and when we compared to the one before, we had 28 or the file before was 28 megabytes. Now it's 15.2, so not really the half, but a lot of reads. No, not a lot of reads. A lot of mapped reads to the reference got rejected, and that also makes sense. I mean, the chromosome of a mitochondrion is quite small, but it's open, so everything binds. All these adapter sequences can get integrated into that. So that's not very surprising. The next step of the analysis would be to get rid of duplicate reads. If you remember the, let's say, the rough or how a PCR amplification works, there might be read duplicates that match exactly, or that would match exactly the same genomic region a couple of times. So we would have a bias that even if it's not part of the measurement, but based on a technical reason of this PCR amplification, we would have enriched regions that are actually not enriched. That's why we want to take out these technical influence. And again, the mark duplicate tool can be clicked, and we take our filtered BAM without the mitochondrial DNA. And what's important here, usually these specific reads are just marked but not removed, and we want to get rid of them. That's why we click here on yes. Then this filter step is automatically applied. The rest of the parameters can be stay as it is. And then let's see how many would get removed. I have to mention that the output of this tool is not that well aligned. So we have some text file, some separated text file, and as a hint, here's a format hint how to transform them in a more real layout so that you have directly the table here. We will not do this now because it's just a transpose of the columns, and then Galaxy displays this automatically. And so this then after this transformation step would look like this. So we have 135,000 read pairs. Remember the 280,000 reads we started. It's way less. 3,500 duplicates we got rid of. An optical duplicate is a specific category that is not that interesting for us, but they are also removed. Below 1%, perfect, and estimated library size looks fine. So, perfect. That was the filtering part. We can now again do a quality measurement to see the distribution of read length in our dataset. Now we are in this part of the workflow. For this, we just create a histogram of all the reads. And the parameters we apply, we take the BAM output of the previous filter step. We set the lower base pair limit to 0 and the upper base pair limit to 1000. I mean, that's the area of the histogram we want to plot. I mean, you can also increase it, but at some point you just don't have values anymore, and negative length doesn't make sense for us. So we take the output, take it to 1, take it to 1000, and we execute it. And the result hopefully looks like this.
a question from my side. Um, would that quality measures you are doing again now, would that can be compared like before the quality measures before and then after?
You mean the histogram? Before, I didn't plot this. We can try it later. Yeah, good. I mean, of course, they can be compared, but I think the distribution would somehow look similar. But interesting, I never plotted the raw data. I can try it later, I'm sure. So, okay, this is the. It's interesting. Here's histogram. Ta-da! So this is the graphic. So we see, what do we see here? We see one peak around 50 base. So on the x axis, we have the base pair length of the reads that are in our data set, and on the y-axis we have the frequency. So, and what do we see here? Nearly 4 peaks. So one at 50 base pairs, so roughly 50 base pairs. One around 200, one around 400, and one close to 600. So what do they mean? The first peak around 50 base pairs corresponds to the TN5 transposase inserted to the nucleosome-free regions. And so these small adapters here, these are the 50 base pairs nucleotides. The second peak around 200 corresponds to small fragments that are inserted around one nucleosome. I mentioned before that the packing structures at nearly 100 or 200 DNA base pairs can cluster around one nucleosome. This is something you see here, because when there's only one element, then of course the distance to the other one is exactly this 200 base pairs thing. The third peak around 400 is when you have two adapter sequences that bound directly to a free part of the DNA. And the sixth is when you have let's say a third transposon that also binds to it. I'm not sure if it's possible to have it in the correct here. So this is probably the 200 peak. So elements that only bind so this peak appears when there's only one element between two nucleosomes. The first one between two, and then that's not shown here. Sometimes you have also three that are shown here. Probably with a bit of fantasy, you see more bulbs here, but that is not of practical relevance. In the tutorial itself, there are also some frequencies shown that did not well work, and that's probably also the most interesting part of this quality control plot. So you cannot use these frequency as for an analysis. You can just see if you can just guess that your analysis or measurement or other reasons cannot or did not perform well when the distribution you receive here is different from the one you actually see. So, for example, you can see this here, there's only one big peak on the bottom, around also 50 base pairs, maybe a bit less, and no other peaks at all. Here you don't have nucleosomes at all. I'm not sure, but it can be that when you just plot the mitochondrial DNA, probably looks like this. Yeah, when you don't see the let's say the fragments or the reads that appear when two of these transposable elements bind, then yeah, the message you get out of your analysis is also somehow meaningless. So the number of peaks is important. Yeah, another one. I'm not sure what went wrong here. It just says that it's noisy. Take into account that the data set you're analyzing should definitely look like this. Probably the peaks are higher or even smaller, but you need to see these 4 peaks beside this. It's not relevant. So in our analysis, we are now done with the pre-processing. So we have the reads that we measured, and we have them annotated to the reference genome. So we know where each read of our measurement maps against the human genome. But what we don't know yet is which of these regions of the human genome are enriched. So in which regions, in our example, most of the DNA is not clustered around an histone, but openly accessible, because this is the stuff that is actually meaningful. Imagine you control 2 samples, I don't know, sick and healthy, and you want to get an idea about the reasons of the different phenotypes, then you're interested in exactly these positions. Which part of the human genome are accessible in the healthy state and not accessible in the sick state, or vice versa? Because this means that transcription factors can bind or cannot bind the DNA, and that part can be transcribed or not transcribed, and therefore the gene is more active or less active. The procedure to identify these areas is called peak calling, and there are 2 tools available in Galaxy that are quite famous. Maybe there are more tools, but these are the ones I know. And the first one is MAX2, and the second one is GenRich. In our tutorial here, we use the MAX2 part. In theory, the other one also performs well. But with ataxic data, it's maybe they changed it, maybe it works well nowadays. I'm not sure about this, but for other data set types where you deal with reads, you can definitely use this also. In our case, only the MAX2 is required. And and then, we are interested in the 3, let's say, events that I mentioned before in the histogram part. What happens if 2 of these transposable elements of the ataxic technique bind within 2 nucleosomes? What happens if only one is attached? And if there are also transcription factors, known transcription factor binding sites in between. This is the stuff we want to analyze now. For this, we again go in our tutorial session. We first need to make a bed file out of the BAM file, out of the marked duplicate data sets, because it's just required by MAX2. think there are not more parameters required.
So basically, Stefan, this workflow is like more or less lots of parameters, and without Galaxy, I'm not sure if that's going to be that easy to do such a kind of parameters thing.
Can be, yeah, can be. And there's also ready-made workflow available. And there was a link at the beginning, I think. So there's no need to click through all of these steps by hand and create your own workflow. There, here. When you work with your own data, there's a workflow available that you can apply. But it's always a good idea to do it at least once, step by step, that you get, I don't know, more familiar with the specific types. Yeah, sure. So here all these steps are also then shown, like input data set that you do the fast QC step, the cut adapt, everything we did now, and with all the specific parameters. Always keep in mind when you use your own data set with that stuff, that these quality control steps are not really part of the workflow because they're not used later on, but definitely do them, because then you see in that moment if something with the data set is, or if the data set is corrupted, or you forgot to filter something, or the data type is not correct. All these small difficulties we have with the Galaxy usage. So we have our bat file here. And now we do the max2, because now we want to see which areas of the genome, or in our case of the chromosome 22, because we only have this subset, are enriched. So what are the relevant areas of the genome that we somehow detected with our analyzers? So we go again in the max2 copy, and this has a lot of parameters. So take care that you click the correct one, and don't forget one, because otherwise the you increase the running time and reduce the meaning of your results. So we have no control file. We have a single and bad file because we merged them already. We take our Homo sapiens as the reference genome size. We are not interested in the model. We want our tabular file output that we can use later on, probably for the multi-QC step. We want peaks, and that's basically it. An interesting part is maybe also that you have the extension site and the shift size as a parameter here. It means when you have, let's say, your read in our cut, adapted, filtered, mapped dataset, we are interested in areas before that read and also in areas after that read. Because imagine you have a interesting site, then probably factors bind closely and not only on the specific position that we increase. That's why we increase it a bit. Minus means backwards extends means a bit further in the DNA. So are you pooling treatments files? No. Do you have control file? No. Single and bun. No appearance
I think single and bed you should select probably.
Sorry, what?
Single and bed, I
Good
suppose.
point. Good point. Thank you. 200, set shift size to -100. And peaks. So that's the output. Peaks is tabular file. This is something you see more and more in Galaxy tools, that you can click additional outputs and or deselect them. This is quite nice to avoid a spoiled history. Peak summits stored in bed graph files, so in advanced. No broad regions. Use more sophisticated. Yes. How many duplicate texts? All. I think that's basically it. That's fine. Should be fine. Okay. I think the tutorial gives another information why it's important to do the mark duplicate steps before. Yep, here. They're doing a PCR, or dealing with PCR duplicates we did before. And now we are dealing somehow with the, we lost the paired information because we somehow merged them. And why this is important is shown here, just as a side note. But perfect. We are nearly done with the preparation, and soon we will have the graphics. So, yeah, the result files itself are not that easily accessible for us. You have a bed graph file of the treatment and of the control. Here's some other bed files, and what we are now interested in to have a nice graphical representation. So first of all, we want to have a nice overview of the areas of interest that are part of a transcription start site. Second, we can check if these areas are located in intergenic regions. And the third one is we want to have a closer look on one specific gene, and see where, for this specific genes, the specific peaks we calculated, where they are located. So these 3 steps will follow now. The point is we have many genes, and we are still dealing with the whole chromosome, so we can focus on a specific part of the chromosome 22. In our case, or not on a specific part, but on specific parts. And for this, we focus on the CTCF DNA binding protein. If you're more interested in the detailed description about this protein, there's a nice paper showing it with a bit of biological background. Interesting, but for Galaxy analyzers, not that relevant. But it's always good to have background knowledge, of course. So we are reducing the size of our dataset by focusing on the CTCF. So first of all, we filter it. We go back to the encode dataset we downloaded at the very first step. We're focusing on chromosome 22. This is everything. This should be done quite fast. Yep, it's there. So we take the output of the previous filter step that we only that we did before. Then we have the one output per kind of input B. Wait. Combined one output for we take the chromosome 2 genes that we also downloaded in the beginning, and what you written the output file. Okay. This one required overlap on base pair. Report only those elements that do not overlap. With file B, yes. Hopefully I didn't forget anything. Should be fine. Execute. See that the number of regions editing is now nearly 4,000, so easier maintainable. The name is a bit confusing, so in the tutorial is proposed to change it. So we have now the intergenic CTCF peaks of chromosome 22. Then the next step is that the tutorial proposes to change it from the output from the MAX2 data format to BigWig, which has some benefits for further analysis. It's quite easy. We take our output of MAX2 bed graph treatments, convert defaults, and also here the name of that result is a bit spoiled, so we should also rename it. Galaxy already proposes here the next tool we should apply is this compute matrix. This will be the next step. And then we will have our calculated heat map. Yeah. Perfect. Run through. We will rename it. So, and now we can come to the plotting part. So what we have now is we have our aligned data set to a reference genome, but only chromosome 22, and we only have regions left that are known for this CF DNA binding protein. And now we want to have an illustration if we have, let's say, around the transcription site some enriched regions. So are these open frames that we measured in front of the transcription factor, behind of the transcription factor, or if they appear in a distance, so for some enhancers and silencers that do not bind directly to the transcription start site, but a bit more distance. For this we use the compute matrix tool, and afterwards the deep tool heat map. We take as an input our chromosome genes, sample order matter no. This GOR file is the max bigwig, so I'm just applying these parameters here. No, sorry, these parameters here. Is it the reference? Okay. Reference point, beginning of a region, show advanced output settings no, show advanced options yes, and convert missing values to 0. Yes, this should do the job. And the next step then is plotting it. So we take the output of the computed matrix because it's a pre-calculation for our plotting. We don't want additional options. And then we click on execute. And hopefully the output is a heat map that gives us an impression where the enriched regions are located around the transcription start site. And let's have a look. Perfect. So what we see here is what we see here is that's around the transcription start site of a gene. We have 1000 base pairs forward and backward. On the x-axis and on the y-axis is some frequency measurement, and what we see is that the reads we measured are slightly before the transcription start site. That's the first impression we have. The second one is that the graph is not symmetric. So on the left side you have a straight, somehow straight line. On the right side it's a bit more loose. And the heat map shows a similar picture. So we have here on the left side the genes, so the several hundred or the several thousand genes, and on the bottom again the transcription start measurement we also have above, and also you see here that the enriched regions in dark blue are on the left side also before the transcription start site, which also makes sense because this is an area of factors that bind that the transcription of a specific gene can start. It makes sense that these areas are accessible. Perfect. The second graphic of interest would be to do the same for intergenic regions, because transcription start site is nice, but we know that other areas of the DNA are also of relevance. So we again go in the compute matrix.
Yes, Sudhakar.
Sorry. Click again on our data set, sample that matters no. We are dealing with our converted BigFig file. Use sample names in the history, okay. Reference point. We now take the center of origin. Show advanced output. No. Here we show yes. Convert missing values to 0. Yes. Just finish the calculation, and then we do the same as before with plot heat map. Show output settings no, show advanced settings yes. So at the moment I'm in this area. The parameters are very similar to the ones before, just that we're now dealing with the intergenic regions. Yes. Change the color map. The color is not really important. You can pick whatever you like. You just want to have a different picture with a different scale than before. The x-axis label is distance from peak header, CTF peaks, reference point labels. Did you hear something? These labels, of course, you can pick whatever you like. So it's done. Execute. I can just say it again, that's quite beneficial if you use these tags, because you then immediately see when you picked the wrong input file. This is in our case not really a problem, but imagine dealing with a couple of data sets that have the same data type, then it's quite easy to, what happens quite easy to pick the wrong file. Yep. So the heat map is also there. Let's open it again in a new tab. So compared to the transcription start site here, where we don't have this symmetric view, and also shifted enrichment to the front part of the transcription start back at the peak center, it's way more symmetric. So it's nearly the middle. You have from both sides some kind of enrichment. What actually this second peak here means, we have to see. Probably it's based on the center. I'm not sure if the result with other proteins look different. Probably the relevant part for us is the area around this peak center. So this is the second graphic, and now comes the most interesting for us, because we want to actually see if there are enriched regions around a specific gene. So now we are having a look on a specific gene, specific data set, and see where the stuff we measured, where the peaks are located, if this is actually relevant. And for this, we use the PyGenome tracks tool. So we are at the moment on the lower, so the deep tools we just did with the heat maps, and now we are on the PyGenome tracks on the right side. And this is then also the last step for today. Again, so with the two heat maps before, we just got a rough idea where the enriched regions of our dataset are located, regarding to the transcription start site or the intergenic regions. And now we want to have a look on a specific gene. So for this, we take the PyGenome tracks tool, and the tool works itself that you can specify specific tracks, and that are then shown under each other. So the first is, so I'm currently adding these parameters here. We want to have a look on a specific gene called RIC2, and this gene is located in that area of chromosome 22. So we have to specify the chromosome, and we include our first track. So the first one is a big track. This is the stuff we created before. We set a plot title, take our big wig file, set the minimum value to 0, the high to 5. This is a question of the plotting axis scale, and show visualization range. Tuck. Yes. Then we pick the second track. This is the narrow peak track. I will explain these tracks in a second. Plot title. Narrow peaks. Display to use box. Plot labels. Nope. This third track is the gene track we are interested in. In our case, this RAC2. Plot title is genes. Format is chromosome 22. Plot labels. Yes. 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 And this is just a question of how you want to display the stuff. You can play around that as much as you want. So this is not a calculation tool, it just creates the image. And then the third one or the fourth one is again a narrow track. Plot title CT F peaks. Our dataset, we take the original bat file we downloaded in the first step. Color, I don't know, blue, and display to use, box, plot labels, no. And we want an x-axis on the bottom to see where we are actually located. Let me check it again. INBIX coverage. Track. Peaks. Narrow peaks. Box. Track. So, look. So the tool just started is not interactive, it just creates a static image. There are tools available where you can also play around afterwards, but yeah, for demonstration purposes, PyGenome tracks is fine. And it should also work quite fast. So hopefully, yes, that looks good. So this is the actual result of the whole analysis we did. So as I mentioned, we added now several tracks, and each of these tracks is shown here. So the first one is the coverage from Max2. The second is the peaks from Max2. The third one is the display of the RAC2 gene. The third one is the CTCF peaks that we are interested in. Remember, this is a DNA binding site. And on the bottom, we have the x-axis. So what does this mean? On the first track, the coverage is somehow the raw data we had, beside all the filtering step and the pre-processing we did. You see that there are specific regions more enriched in our data set than others. So these are the, let's say, high peaks. This is the output of the Max2 peak calling step. The second track, this is and the second track is, let's say, the Max2 algorithm identifies areas that are extremely high enriched in our data set. These are the 4 buttons or 4 blocks here. So here one, the second one, the third one, and the fourth one. The third track shows known transcripts of the gene we are interested in. So we picked the RAC2 gene, but we increased a bit the size of the area we want to investigate our data set. That's why a second gene is also shown here, or the transcripts of a second gene are also shown here. What you see here is that the transcription site on the beginning of this RAC2 gene slightly differs, different transcripts, so not that surprising. And that the peaks from the Max2 peak calling event is, at least in this area, directly located in the transcription start site area. The second one, not really. And then you have a third one that is in between of 2 genes, and the third one on the back side here also. Here it's quite, this stuff needs to be investigated further. It can be that there are known chromatin factors that are relevant for a specific position. It could be that there's histone modifications that are known. It could be that there are methylation sites that are highly enriched in that area where other binding factors can also bind. For this, then you can have a look on the other tutorials of the epigenetic category, and then investigate this area more. It just gives a rough overview. Okay, there is something. Now we can go deeper into the topic. And the last track, not that I forget it, is the CTFC peak track. Remember, this is the DNA binding protein. We know that specific binding sites of that protein are these are known, and we can now see if the peaks we investigated with our Max2 algorithm are somehow located next to this protein, or next to the binding sites of this protein. And the first one, not really. This intergenic region, probably. And also on the back side, could be that there's some kind of correlation. Yeah, that's it. But even here, you have to investigate it more in detail. I mean, it can be that you increase the scale, have a more detailed look on specific positions, check databases of known binding factors. So the amount of possibilities is very large. a static picture cannot answer this question, but it roughly gives you an idea about the area where a closer look is maybe beneficial. Yeah, exactly. So let me check if I forget something. No. So when you have another look on our analysis scheme, we are done with all of these steps. And now for a specific gene, a specific area of our human genome, a rough overview of the data set we applied could have meaning, maybe not. This is then part of further investigations. So to sum it up a bit, we somehow did it in the first try, or I guess you did it in the first try. You get a rough data set, you have to do some filtering, trimming of the nucleotides that are part of the technique and not meaningful for our results. You need to get rid of mitochondrial DNA, you need to get rid of duplicates, you need to get rid of low quality reads, too short, stuff like this. And after all these pre-processing, you can ask a peak calling tool like Max2. And as I mentioned, there are more also available for enriched regions that might be a good idea to have a closer look on. And then you can use deep tools and PyGenomeTrack, for example. Also here are plenty of more tools available to get a general overview about the location of the enriched regions in relation to a transcription start site or intergenic regions or any category of your choice. Or you can have a look directly on a specific gene, also start and middle, where peaks appear, why they appear, which binding factors actually can bind to it, and then have a rough idea if a specific gene, when you detect the difference in healthy and sick, can play a role or not based on ATAC-SEC. I mean, there are plenty more tools available, or techniques available, but this is the message from that. Yeah, I think that was everything from my side.
Great, Stefan. So that was an extensive session, showing up each parameter and everything into the epigenetics to recognize, to basically access the chromatin area and to be able to, let's say, gene expression, so control gene expression. So the really good session. I hope people had a good view of how it could be, Galaxy could be useful in such a huge steps. But yeah, I would keep the session open for a minute or two if there are any questions.
I mean, the general point is definitely that if you want to get deeper into that topic, just do it by your own, click it, play around with the parameters. I think in the session before it was also mentioned, it's always a question of practice to get a feeling which parameter has which influence, which questions can be answered with the technique, which not. Maybe there are other tools. Even if you miss a tool in Galaxy, you can ask for it. And, if somebody has time, they may be added to the tool sheet and can be also applied.
Yeah.
It's always practice. Yeah, exactly.
So right now, if you, okay, yes, okay. So let's take a group photo, please. Meanwhile, if you any of you have any questions, you can post by the time we take the photos.
Should I
Yes,
stop
you can
present?
stop
Yeah.
the sharing. Yeah, that's fine. Thank you.
Yeah, please, everyone.
Kiran,
Yeah, please go ahead and show.
yeah, for some of us, the camera is not enabled.
Oh, okay, just a minute. Okay.
Yeah, please,
Yeah, sorry.
yeah,
Yeah,
please, now you
let's.
can do
You can try now.
Thank you.
And we will try now. I just re-enabled again. Okay, is still some problem with the cameras?
I don't think so. It is like it's in active mode, I think. Everyone can join, please.
So it would be great to have as many as cameras because it's the last session.
Yeah, for this day.
Okay, I will take a picture from here. 3, 2, 1. Thank you.
Yeah, with your permission, I would like to
stop the recording. Yeah, that's it. Yeah.
Yeah, thank you, Stephen. That was a very exhaustive and detailed explanation from your side. I really appreciate from the program committee. Now, as a part of the event, here we comes to closing our pre-conference workshop as a part of INBIX 2021. So, regarding the closing remarks, I would like to talk a few lines about Dr. Kiran Telukunta, who is our backbone for this workshop, pre-conference workshop. I would just like to brief about Kiran. Kiran did his Masters of Science from Albert Ludwig University in Freiburg, Germany, on artificial intelligence and bioinformatics. And thereon, he moved for doing PhD in Albert Ludwig University again in pharmaceutical bioinformatics. Thereon, in between, he did part-time jobs like in software engineer, scientific cloud coordinator. Then he worked for international system assistant. Then currently is working as a cloud architect. Apart from that, Kiran, has got its own. He's one of the directors of TMS Foundation, which basically he runs this. And then he also works for BioClose, working for Galaxy, getting to bring to India. So Galaxy BioClose India with the help of TMS Foundation. So he's been worked on several databases, have technical and natural science background in pharmaceutical bioinformatics. So he has done a lot of projects, and he did some master courses at different universities in Germany, and is voluntarily prepared the submission of platforms for online workshops. During the workshops, he plans to present some general guidelines and underlying principles for gene clustering analysis using the applications of Galaxy. With these few points, I would like to call Dr. Kiran Telukunta to give his closing remarks. Yeah. Open to you, Kiran. Thank you. Yeah, please, Jaravan sir. Yeah, please go ahead.
Before Kiran gives his final remarks, I would like some comments about the workshop from a few of the participants. Yeah.
Yeah, that'll be
So
wonderful, sir. Yeah, please.
Yeah. From some participants. Yeah.
Yeah, thank you, sir. Yeah. Yeah, please feel to volunteer to talk. Okay. Before we going further, there is a question from Nanda Kumar. Thank you for the session, Stephen Fleming. As you know, that epigenetic factors are denoted by alterations in DNA methylation, histone modification, and transcription of regulatory non-coding RNA, or these additional epigenetic changes that takes place do not involve these mechanisms? Stephen, I think if you're able to got the question, that's fine. Otherwise, you can just see the chat box. Yeah, thank you. Stephen, are you on?
Yep, what again, please? I was drinking something. Can you repeat the question, please?
Yeah, it's there in chat. Otherwise, I can repeat it once again.
Ah, okay, sorry.
Yeah, no problem. Thank you.
Okay, sorry, yeah, Brad. The question is, so at least the mechanisms that can be analyzed in Galaxy are DNA methylation, histone modification, and mapping of DNA knowledges or of database knowledges that is of known databases that can be mapped to the data set you want to analyze. Are there more? The question is what counts to epigenetic stuff. I think the definition is not, or is changing over time because more and more factors are developed. Small RNAs are also sometimes taken into account. But I think good questions. I can look it up. I don't know. These are the ones I had to deal with in the past, let's say.
Thank you, Stephen. Yeah. I think Nandan, you can write a mail to Stephen. We'll provide you the email ID. Yeah, you can feel free to contact Stephen. Yeah. Thank you, Stephen. Thank you, Nandan, for the wonderful question that you asked. Yeah. Thank you. So over to Kiran.
Okay, great. So thank you all. So at first, the very happening of this Galaxy workshop under the banner of Galaxy India to the Indian audience, along with the renowned INBIX conference, is one of the milestone, I would say. So this is what at least we came to this position right now. After last year, after the GCC 2021, where a soft launch happened. And if you see in this conference, almost 40% registrations during the INBIX conference opted for the Galaxy workshop. So which indicates the interest in the Galaxy framework, in the natural science research, and also in the industrial community. Opting open source Galaxy for framework is like to stimulate the research is in a way which suits for India also, because it's an innate nature for India to actually share, do the things. So that's very much easy way for Galaxy framework to get spread into the India, I would say. And as Shastri sir also pointed out, the main objectives are to, like, of course, it is a free open, having Galaxy is a kind of a free source, but not just that. The second thing is like making changes, easily adapting to the new innovations without effort, will be an effortless method with the Galaxy. And this will be very useful. So these are the two main aspects of the open source, as Shastri sir pointed out. And strengthening of the open source drug discovery is a kind of theme this year in the INBIX, which was strongly supported by the keynotes of Chris and workshops of Simon and Nagamani Hriday team, which covered and presented like chemical toolbox in a web server for processing, analyzing, and visualizing the chemical data, and also performing molecular simulation as part of structural bioinformatics and chemoinformatics using Galaxy. This was done by Chris, and subsequently with the workshop presented with the protein ligand docking, which presented a much more deeper understanding of the practical analysis by Simon, and a pool of MPDS tools shown by Nagamani Hriday team for tuberculosis, for diabetes, for metabolic disorders, COVID-19. Of course, this makes much more useful in the Galaxy itself. COVID-19 Galaxy project is a very popular one, and the COVID-19 MPDS tool definitely will be a kind of combo useful for research in that direction. And more are coming up in the MPDS tool. So this is kind of a strengthening in this direction for the drug discovery, which was very much less in before in the Galaxy way. So that was very much an improvement into the Galaxy side. The other workshops, as usual, and the keynotes continue to shine different areas which Galaxy addresses, such as drug resistance determinants, omic areas, and epigenetics. Participants had the opportunity to listen to know how other parts of the world use Galaxy, in particular about Australia, where Gerath presented about it, and also he expressed about the use Galaxy star other instances, how they are working, and about the tri-yearly updates of a code base. So it makes very much important for the people who are from the background of computer science, or let's say programming. Tri-year updates will be very enthusiastic for them to try to attain the milestones and get to an updates. And every update is a very actively celebrated in the Galaxy community. If you happen to be in the Gitter communities while happening these Galaxy updates, they are very much excited on every update. And a new popular updates in Galaxy was discussed by Gareth, and Galaxy tool community, how it is available and where it is available, or methods of availability, contribution to the COVID-19 Galaxies, about AMR and its detection with the Galaxy. The workshop by Dr. Vipin was very much useful in that area. An extensive proteogenomics workshop with database creation, database search, and novel peptide search, showing up each and every options available and explaining in detail by um, Sona Charles. An introduction to epigenetics workflow showing the ability to access the chromatin and also subsequently regulating the gene expression with several parameters, which has shown why the Galaxy could be very useful, where 100 parameters have to be changed. So this was a kind of an example for us. It could be why Galaxy could be useful, and you redo the things, and maybe you can experiment. So that is a kind of an example for that. In the end, I would like to thank all the speakers, Shastri sir, Gerath Price, Dr. Anshu Bharadwaj, Vipin Singh, Dr. Vipin Singh, Dr. and Gerath Price, in particularly, doubly thanks because he in timely replaced Dr. Dave, who could not make it to the workshop, and Dr. Christopher Bernard, Simon, Dr. Nagamani, and the chairs, Sona Charles and Dr. Stefan Fleming, and the chairs, Dr. Jayaraman Valadi, Dr. Harpreet Singh, Dr. Prashant and Surav Janala, Dr. Sangeeta Sawant, Dr. Sugunakar Uri, Dr. Anil Kumar, and for Dr. Anshu Bharadwaj, timely replacing the chair, which we were not having. And thank you very much. So, with the help of the Galaxy Workshop committee for this INBIX conference, Dr. Harpreet Singh, Dr. Titraj Singh, Dr. Sugunakar Uri, and Dr. Prash Surav Janlala, we made an effort to get all the people to on board and to have a successful workshop event consisting of 3 keynotes and 5 workshops. Um, so, and the chair and the NEAST technical team who managed all the sessions to go very smoothly, thanks to them. And yeah, in the end, thanks to all to make this workshop very successful. But this is just the pre-conference workshop. You should remember the conference is going to happen in next 3 days, the INBIX conference. Hope most of you already registered in that. Who opted for Galaxy Workshop mostly opted for the INBIX conference also. If not, please do that. And in the end, I thank everybody. And also, please look for your mail. You will be receiving a mail for the feedback form so that we can know how we performed and how we participate, how we can improve the participation, and how the sessions can be improved, what kind of other resources you might require. So such a kind of things in the feedback form we will like to ask you. Please actively give your feedback so that we can understand your interests and requirements. So thank you, everyone. Thank you for coming to the INBIX pre-conference workshop. So that's all from mine. So Sugnakar, is there?
Yeah, thank you, Kiran. So I look forward for all the active participation from tomorrow. So we can have some brainstorming sessions starting tomorrow at 9 a.m. Yeah, 9 a.m. starting INBIX conference. Yeah, thank you, and all. Yeah, if any questions are there, please feel feel free
Free
to
to
say
put it
back.
into the guitar.
Yeah, yeah, yeah.
Yes.
Yeah,
Okay,
please
thank
don't
you all.
forget to give the feedback. Please don't forget to give the feedback. That's quite important. Yeah, thank you. Thank you, one and all.